its application. On behalf of the organizing committee, I welcome you all. And we have amongst us three very distinguished speakers. And for that, I request our Principal Maharaj, Shami Kamala Sanandoji, to formally deliver the welcome address. On behalf of the organizing committee, I welcome you all. And we have amongst us three very distinguished speakers. And for that, I request our Principal Maharaj, Shami Kamala Sanandoji, to formally deliver the welcome. On behalf of the organizing committee, I welcome you all. And we have amongst us three very distinguished speakers. And for that, I request our Principal Maharaj, Shami Kamala Sanandoji, to formally deliver the welcome.
Is there any like a time lag?
Yeah, there is a time lag.
So just after my welcome address, Dr. Manna, then we can start, sir.
Yes, sure.
Time lag?
Yeah, there is a time lag.
I think somewhere there is an echo.
I think
It's kind of an repeating.
time lag.
Other than who is speaking.
What we can do, like during the presentations, like except the speaker, the others, like most of the others can like switch off the video mode. Only the audio mode will maybe that time streaming will be much better.
And also, I mean, if you're not
Microphone.
using the mic, then it's better to keep it mute.
Micro and video both off.
Then we can just.
During the presentations, like except the speaker, the others.
Mute this. Yes, yes. Yes, then the noise do not come. Yes, yes.
And also,
Microphone.
the mic, then it's better to keep it
Micro and video both off.
Then we can just.
So Dr. Manha, yes. So today we have organized this one-day national webinar on chemistry and its application. In this webinar, we have got three distinguished speakers. Number one speaker, Dr. Debasis Manna, Professor, Department of Chemistry, IIT Guwahati. Second speaker, Dr. Atikur Rahman, Assistant Professor, Department of Physics, IJR, Pune. And third speaker, Dr. Niladri Patra, Assistant Professor, Department of Chemistry, IIT Dhanbad. And we are very fortunate that three learned speakers are present in this webinar, and they will discuss various features of the chemistry and its application. I request all the members, please, I think some mute the microphone.
মহারাজ আপনার যে স্ট্রিমিংটা রয়েছে ওটাকে সাউন্ডটা মিউট করুন। আপনার যে স্ট্রিমিংটা হচ্ছে ওই সাউন্ডটাকে মিউট করে দিন। যে স্ট্রিমিংটা রয়েছে ওটাকে সাউন্ডটা মিউট করুন। আপনার যে স্ট্রিমিংটা হচ্ছে ওই সাউন্ডটাকে মিউট করে দিন।
Now it is okay.
মহারাজ আপনার যে স্ট্রিমিংটা রয়েছে ওটাকে সাউন্ডটা মিউট করুন। আপনার যে স্ট্রিমিংটা হচ্ছে ওই সাউন্ডটাকে মিউট করে দিন।
I think now okay. So this today's discussion, especially our esteemed speaker, they have selected this starting from the biomolecules and ultimately goes to the different nanoparticles in the present day. These biomolecules, especially enzymes, are also nanoparticles, but it is all are biomolecules. And these nanoparticles in a globular form in body, it has several roles and its complex function. And it is also yet to reveal to human beings whole activities of this nanoparticle as enzymes or globular protein in body. And afterwards, this nanotechnology and its application in energy harvesting, especially the self-cleaning purpose. Self-cleaning means it is very interesting thing that renewable energy, especially solar energy, it actually requires the solar panel, and all at present all are made of silicon. And the silicon made of this solar panel, when fixed in the roof or otherwhere, and all times actually it exposed to environmental dust particles. And 3 or 4 years before, when we have installed our college this on-grid solar system, I was invited in a panel discussion. There are many people from India and abroad also present. I am also a lay person because I am not my subject is not energy management. But they asking the experience of myself regarding the solar panel and everything. So I told as a lay person, as my experience, I told that main problem of the solar panel in especially urban region, it's cleaning problem, because we facing this, that if we sprinkle the water and also forcefully sprinkle all this, but dirt particle could not remove easily. And sometimes after 2 or 3 times washing, again when it will dry, again this dust particle still sticky. So I told that dust particles, these are actually accumulated in solar panel, which is actually nothing but charge particles. So it is difficult to wash this only just soap water or water. What we are facing this problem. So if anybody thinking some solar panel which is self-cleaning, then it will actually solve the problem. Then after when I come down after panel discussion to the scientists of German University, they are also present in this webinar. Oh, sorry, this panel discussion, the participant group. They told that they are working in this field, within few years it will come in. But nowadays it is coming also, and our second speaker, he will enlighten regarding self-cleaning in this energy management where nanoparticles will work. And the third speaker is also talking about this self-assembly of nanoparticles. It is very important thing, which is not only important in the electronics, but also important in the field of drug delivery, design, cell imaging, and all this. So it is a very important application of chemistry what we are dealing today. So we, on behalf of this college, on behalf of the all faculty members, students of chemistry department, we heartily welcome our three distinguished speakers from on behalf of this college for their consent for delivering their talk through this webinar. So also I tell the participants, please heartily participate in this session. After each session, there will be a question and answer session. You write all questions to the chat. After that, all questions will be addressed after each speech. So with these few words, once again, I extend my welcome and regards to the distinguished speaker, and also the thanks to the all participants for joining this webinar.
Thank you, Maharaj, for your welcome address. So, as you all know, that there will be 3 lectures, each with duration of approximately 35 minutes. And after each lecture, there will be a question answer session for approximately 10 minutes. So I request all the participants to type your questions in the chat box, and Dr. Supratim Shui of our department will collect those questions and forward it to the distinguished speaker on your behalf. So in this technical session, our first guest is Dr. Debasis Manna from the Department of Chemistry, Indian Institute of Technology, Guwahati. And before inviting him to deliver the lecture, I request Dr. Chandrakanta Bandyopadhyay, Head of the Department of Chemistry, to formally introduce our distinguished speaker. Over to you, sir.
Respected distinguished delegates and respected speakers and my dear friends, really it's a pleasure and opportunity to introduce such eminent speakers. Here today, we have our first speaker, Professor Debasis Manna from IIT Guwahati. He did his BSc from Ramakrishna Mission Vivekananda, Ramakrishna Mission Narendra Residential College, and then he went to IIT Bombay for his masters. Then he did his PhD from University of Chicago, USA, with Professor Cho on biological chemistry, and did his postdoc in Calcutta, in Bose Institute with Professor Gautam Basu. Then in 2009, he joined the Department of Chemistry, Indian Institute of Technology, Guwahati. And there he started his own research. He has many awards and fellowships in his credit: Young Scientist Research Award, Young Investigator Awards, etc., from different institutions. He has published many publications, around 45 publications in different reputed international journals, and he is awaiting a patent also. And his research interest is on the drug design in cancer immunotherapy and development of synthetic anion transfer for treatment of different channelopathies, including the cancer. And today he is going to deliver the lecture. The topic is indolamine 2,3 dioxygenase inhibition as a strategy to augment cancer immunotherapy. With these few words, I would request Professor Manna to kindly deliver this lecture. Professor Manna, please.
Okay. is it shared? The screens, can you visualize?
Yes, yes, we can hear you.
No, can you visualize my screens?
Yes, I can see it.
Okay, okay. So thank you so much, sir, for your kind introduction. And thank you all, Principal Maharaj, for giving me the opportunity to talk about our current research works. Respected teachers and dear students, today I'll be talking on inhibitions of indolamine 2,3 dioxygenase as a strategy to augment cancer immunotherapy. Okay. So before I go and talk about our research today, so I can to introduce ourselves here. We work on chemistry department, Indian Institute of Technology, Guwahati. Our main focus of research is development of drugs and drug carriers to induce controlled cell death. Okay, so we mainly focus in 4 different research area. One is like in understanding how lipids interacts with the proteins to have an idea about their involvement in drug developments. We also works on lipid-based drug carriers so that we can develop and stimuli responsive drug carriers to develop synthetic drug molecules or commercially available drug molecules, particularly to the cancer cells. We also work in the transmembrane ion transport and diseases. So I just a little bit want to elaborate in this research area. This is quite a new, we have just recently started on working on that. It is in the field of targetless drug discovery approach. So why I said targetless drug discovery approach, in generally in drug discovery, we always have basically a target, either it's a protein or it's an enzyme or it can be in genetic materials. But here what we do, no, we don't need it, targets. What is actually we're working on here, we want to actually perturb the ion homeostasis across the membrane bilayer to basically control, to induce the controlled cell death or have an other implication in other diseases. But today I'm going to talk about drug discovery and cancer immunotherapy. So here what I'll do first, I'll give a brief overview about cancer and what is cancer immunotherapy and why IDO or indolamine 2,3 dioxygenase. In briefly from now on I'll be talking about an IDO can be considered as a drug target, and then I'll just give us few examples of our previous and ongoing research work in this field. Okay, so for those for those students who are actually not aware of what is cancer, but for the who knows, you can ignore this part. But for those who are completely not aware of cancer, I want to say that the cancer is a multiple disease where cell grow out of control and invade and destroy normal cells. Okay, so in normal body, cell grow, divide and die in an orderly fashions. But tumor cells basically is different because they do not die. They just continue to grow and divide in a disorderly fashions. Tumor cells may become cancerous due to the accumulation of defects or mutations in their DNAs. So certain inherited genetic defects or infection can increase the risk of cancers. Obviously, the environmental factors like air pollutions or our lifestyle, such as smoking, consumptions of heavy alcohols, can also damage DNA, which can leads to cancers. Most of the time, cancer cells, whatever in our body, can repair these damages because we have this DNA repairing enzymes which can repair basically the damage what is done in DNA levels. However, if a cell severely damaged which cannot be repaired, okay, so then in that cases those cells basically bypass the apoptosis or programmed cell death process and leads to the cancers. So sorry, yeah, now it's moving. Okay, so I said that the cancer cells, it grows. Okay, So here, one thing I want to clarify that when we talk about the development of the cancer cells, we should be a little bit careful in defining the cancer cells. Okay, so when the cells multiply abnormally, we generally call tumors. Okay, these tumors can be of two types. Either is a benign tumors or can be a malignant tumors. In a benign tumors, they are actually non-cancerous. Most of the time they are non-cancerous. They just divide abnormally, but it cannot spread invasion or cannot metastasize to cause cancers. Whereas the malignant tumor cells, it actually invade the neighboring cells, enter the blood vessels, and metastasize to different organelles in different parts of our bodies and cause the cancers. Okay, so that was I was showing about. It is called the metastasis. It originates and then enter into the blood circulation systems. From there, it spreaded different organelles of our body, and where that is basically the last stage of cancer, where we cannot do, most of the time we cannot do anything. Okay, there is no cure for that. So for cancers, early detection is very important. Early detections and proper treatment is very important. So far there are 200 types of cancers, including like brain cancer, colon cancer, blood cancer, breast cancer are few of them. According to World Health Organizations, there are 18.1 million new cases in 2018, and 9.6 million people estimated to have died just because of the cancer. So like every 1 out of 6 global deaths is because of the cancer. However, good news is that 1.5 million cancer deaths were avoided in last 2 decades. So the treatments are obviously there. The treatment options depends on the cancer. It depends also its stages. It also depends on its spread and also the general health of the patients. But the goal of this treatment of the cancer is to actually kill only the cancerous cells, not the normal cells, so that we can reduce the side effects. Okay. Currently, there are 3 strategies or 3 therapies are available to fight against cancer. One is surgery, second one is chemotherapy, and the radiotherapy. In surgery is very common. Wherever there is a cancer there, that organ, we can chop it off from our bodies and we can just throw it out. Okay. Chemotherapy, what we can do, we can actually use organic or inorganic molecules, okay, which can kill or which can induce the programmed cell death in cancerous cells, so that it can bring back the cancerous cell to normal cells and it can actually die. In radiotherapy, what we can have, I can have X-ray, and we can use the X-ray to kill the selected cancer cells. But all these 3 strategies face a problem of the side effects. So what is side effects? Side effects is basically, you have to kill the cancer cells, but during these 3 strategies or the treatment procedures, we cannot selectively kill the cancer cells. We actually also kills the normal cell, and that killing of the normal cells sometimes may also induce the cancers or other diseases. So that is called the side effects. Okay, so now in today I'm going to talk about the immunotherapy. As you know, so by now from my discussion, the cancer is not a simple thing to study about, because cancer cells are very smart. You develop a drug, it modifies after sometimes and survives. So cancer cells is basically creating an war, as we call as a molecular war against the immunocells. Okay, so now we need to understand what is the immunocells, how immunocells works. Because if you think about, our body already has an immunosystem in place. We have studied about the white blood cells. That is one component of our immunosystems. There are actually T cells, macrophages, dendritic cells are also the part of these immunosystems. So what is the problem in cancer? That when the cancer spreads or cancer appears, it actually bypasses our immunosystem, so that it puts our immunosystem in hibernation mode. So our immunosystem basically remains silent, it cannot work. So one strategy is that if we can rejuvenate our hibernated immune systems, okay, then our body itself can fight against these cancerous cells and actually remove those cancerous cells, okay. So, and this cancer immunotherapy has become so popular in the last decade, and it is considered as one of the biggest breakthroughs since chemotherapy. And immunotherapy and traditional chemotherapy or radiotherapy can get benefited from the combinatorial strategies, again, tumor-induced immunosuppressions. So for their seminal work on cancer immunotherapy, Professor James P. Allison and Professor Tasuku Hanjo was awarded Nobel Prize in 2018. Okay. So now let's try to understand how our immune system works. Then our understanding on immunotherapy will be much more clear. Okay. As I mentioned, white blood cells is not only our immune system. There are T cells, macrophages, dendritic cells, and others are there. Also natural killer cells also there. So T cells, one of the leading fighter against the fighter of our immune systems. You know, the T cells you can see here, just a minute. You know, here the T cells you can see here, it has actually a sensor we call PD1, or it's called programmed cell death receptor. This is a surface protein, programmed cell death receptor. I repeat, programmed cell death receptor. So the T cell contains programmed cell death receptor. You know, some of you guys may have seen metal detector. So what metal detectors do or does? It's basically search around us. Whenever there is a certain species metals, it gets a signal back, right? And then we can identify that metals from the field. And that's exactly how T cell works. T cells moves around, okay, with this PD1 receptor in its surface. So if we see some foreign cells like in microbe cells, if you don't get a signal back, so what it do is basically release toxin and kills the microbes. But when it actually see a normal cell give the signal back to the T cells because it has this PDL1, or is called programmed cell death receptor ligand 1. Okay, so that a normal cell has that ligand in the surface. So they communicate. So when they communicate, they find a signal back, and then there is a tolerance. So the T cells don't destroy our body's own normal cells. Okay, so I think now it is clear that only foreign cells which don't have these PDL1 receptors, it can be killed by the T cells of our body. Now the problem happens when T cells meets the tumor cells, and it asks, are you one of us or a foe? But the T cells, tumor cells, as I said, are very smarter. They actually makes fake PDL1 receptors in multiple number, more than these normal cells in their surface. And they keep on giving back the signals to the T cells. So that's how what it does basically, it actually suppress the activity of the T cells and bypass the immune system and keep on growing. Okay, so now the hibernation happens. Unfortunately, tumor cells are parasite. It's not one of us. So the PDL1 of the tumor cells are fake. So we need to destroy it. Okay, so there is basically, if you think about here, it is a kind of a checkpoint. The PDL1 interaction is kind of a checkpoint. Now, what can be done? What basically James P. Allison did it. He developed monoclonal antibody, like a PDL1 monoclonal antibody, so which can actually binds to the PDL1 of the tumor cells, and then basically this PD1 will be free and T cell will release the toxin and will destroy the tumor cells. Okay, so currently in last 6 years, a dozen of FDA Food Drug Administrations approved the immunocheckpoint inhibitors like pembrolizumab, nivolumab has been approved, and these are basically these immunocheckpoint inhibitors are nothing but these monoclonal antibodies and very successful in treating particularly the lung cancer cells. But the problem happens there that these immunocheckpoint inhibitors are not that successful in other cancers like in breast cancer cells. Okay, so what happens again, the tumor, particularly in case of a breast cancer cell, even if you have these monoclonal antibodies or these immunocheckpoint inhibitors, the tumor relapse, the tumor recurrence again. Okay, it appears again, basically tumor spreads. So again, tolerance there. So when this research on these immunocheckpoint inhibitors was going on, in the same time, in late 90s and early 2000s, the search for another immunotherapy was going on from another directions. Okay, so that was basically the enzyme based. Tryptophan is one of the amino acids. Okay, so the tryptophan, one of the amino acid, it is called also the essential amino acid. Why essential amino acid? Because our body cannot synthesize this tryptophan. Okay, we have to get this tryptophan amino acid from plant sources. Okay, and this is very important for protein synthesis. And this tryptophan can be also catabolized either by serotonin melatonin pathways or kynurenine pathways. So I will be focusing on the kynurenine pathways. These kynurenine pathways, the catabolism of the tryptophan produces kynurenine, 3-hydroxykynurenine, kynurenic acid, quinolinic, NAD+, and other metabolites. And these chemical conversions, if you see the structure here, this is the structure of the tryptophans, and this is the N-formal kynurenine. And so this single step conversion, so that basically the cleavage of the C2C3 double bond, okay, to N-formal kynurenine is controlled by indolamine 2,3 dioxygenase 1 or IDO1, IDO2 is subclass, and tryptophan 2,3 dioxygenase. So tryptophan 2,3 dioxygenase present in liver, where it catabolizes excess tryptophan. The IDO1 and IDO2 are ubiquitously present. Particularly, they are actually present in immunoresponse cells. Okay. However, excess catabolism or excess metabolism the tryptophan produce over several metabolites of kynurenine pathways, including like a quinolinic acid, kynurenine, and picolinic acids. And these metabolites inhibits the T cell and natural killer cell proliferations. And T cell and natural killer cells also get inhibited by the depletion of the lower concentrations or depletions of the concentration of tryptophan. So IDO express ubiquitously, as I mentioned, because it's mRNA there all throughout body. But the expression of IDO1 increased or induced when there is a disease condition. So under pathological conditions or wherever there is an inflammation, the stress conditions, in presence of like cytokine like an interferon gamma, the IDO1 get expressed via NF-kappa B or JAK-STAT signaling pathways, in particularly T cell, dendritic cell, macrophages, and other immunoresponse cells, and which all results in basically the hibernations of the hibernation of the activity of the T cells. I'm not going more detail about the biology portions of this activation and deactivation of the T cells. So a variety of studies with the cancer models suggest that the IDO assist cancer progressions and metastasis. So inhibition of the IDO activity improves the efficacy of the therapeutic vaccinations, immunotherapy, chemotherapy, also radiotherapy. And IDO also was reported to be overexpressed in several other diseases, including Alzheimer's diseases, HIV encephalitis, and including the age-related cataract, and wherein also any disease which are related to the kynurenine pathways. Okay, so now let's go back and try to understand our immunotherapeutic model again back. Okay, so what I said that previously, that when you actually develop molecular antibodies like monoclonal antibodies like in PD-L1 or pembrolizumab, you can block these PD-1 interactions with an PD-1 so that T cells can kill. But if in absence of there, it is a tolerance there. Also, now I am going to add it there that in tumor cells also that IDO expressions goes high. So these higher expression levels also allows the tumors to relapse back. So the tumor again comes back and spreads. Okay. So what can be done? We can inhibit the IDO activities. So when it has been seen, when we inhibit the IDO activity in absence or in presence of these monoclonal antibodies, again the T cells successfully kill the tumor cells. Okay, so the conversions of tryptophan to N-formyl kynurenine leads to the immunosuppressions, and this overexpression of IDO level plays a major role. So we need to control the overexpression of IDO1. How can be done? It can be done three ways. Either we can block its expressions, we can also, by genetic levels, we can block its expression, we can silence it, we can actually regulate effector pathways, either upstream or downstream effector pathways. But the problem is that, you know, these enzymes, when we will actually block it in either genetic level or upstream downstream pathways, it can cause other diseases. So as a chemist, what we can think about, the simplest way we can think about, can we inhibit the activity of IDO1 enzyme? Okay, and that, while we started research in 2012, in the meantime, while you see in 2015, crystal structure of IDO get resolved, which actually helped us enormously. So this is the part of the crystal structure of the indolabin 2,3 dioxygenase. If you look at here, this is an enzyme and it contains the heme as a prosthetic group. Without the heme, it is a simple protein and it will not show any activity, like conversion of tryptophan to N-formyl kynurenine is regulated by this heme group. And it contains two binding sites. The cyan color I have highlighted here, it is an hydrophobic binding pocket, and this yellow color I highlighted here, it is an hydrophilic pockets. So if somebody wants to develop an molecules which can bind and inhibit the activity of IDO1, he should think about 4 criteria. One criteria is that it should have at least aromatic ring with 2 cycles, so that it can actually fill in the pocket A, and it can have an atom with a free electron pair, such as oxygen, sulfur, and nitrogen, which can interact the heme iron, and it can have groups in larger ligands can like involve in van der Waals interactions present in pocket B, and also it can have residues which can be involved in hydrogen bonding with the heme 7-propionate groups. Okay, so these are the basic criteria to design molecules. Okay, so before I proceed further, I'm going to talk about the chemical reaction mechanism, how the conversion of tryptophan to N-formyl kynurenine happens. Okay, so first, the heme group, as I mentioned, the prosthetic heme group is very important. So for this enzyme, there are 2 substrates. One substrate is oxygen, and the second substrate is the tryptophan. So the first oxygen binds to the iron and form superoxo anion intermediate. And this superoxo anion intermediate can form peroxo transition state either via radical reaction mechanism or via ionic mechanism. And it can lead to either dioxygen intermediate for membered ring, or it can form an epoxides, okay, which further hydrolysis, autohydrolysis can give you N-formyl kynurenine. Okay, I hope this reaction mechanism is clear, because I'll be heavily focusing on this reaction mechanism in the slides we'll be discussing later. Okay, so what is our goal? So from this discussion so far, one thing I think you should try to understand, or you have got the message that our primary objective is to develop a molecule, an organic molecule, which is non-toxic in nature, which will not kill any cells, neither cancerous cells nor normal cells by itself. What it will do then? It will basically, it will rejuvenate our immune system, and then our immune system will basically kill the cancer cells and regulate the rate of transformation of L-tryptophan to N-formyl kynurenine. So with this approach and with the crystal structure in our minds, we will talk about our first examples. So if we look at here the crystal structure here, one thing I forgot to mention about that there is a histidine residues at 335 positions, which also coordinate the other side of the heme group with that iron. Okay, so we thought about, can we have, can you make a 2H123 triazole molecule synthetically, which contains that lone pair and that can easily interact with the heme iron. So we thought about making an 4,5-diaryl substituted triazole molecules which can fit within this pocket nicely. Okay, so how we can make these systems? Okay, we can make this triazole either through 3+2 cycloaddition with alkyne azide system, or we can make it from 3+2 cycloaddition reaction from tosyl hydrogen, simple tosyl hydrogen. So we successfully developed a reaction methodology, a simple from benzaldehyde we can make a tosyl hydrogen, and this tosyl hydrogen and acts as an amphiphilic reagent. And these amphiphilic reagents either can have an N nitrogen nucleophile or carbon as a nucleophile. So either a polar reaction mechanism or 3+ cycloaddition reactions, we showed that through polar mechanism it cyclizes and forms a 2H1,2,3 triazole in under mild basic conditions. So we made a, with this our developed reaction methodology, we made a series of compounds, okay, but we failed. We saw no significant inhibitions. So we went back to the structure again. What is the problem? This is the crystal structure right over here. What was the problem? The problem is, as I mentioned here, pocket A is hydrophobic. If you pay attention to here, pocket A is hydrophobic and pocket B is hydrophilic. So what we did it with this reaction methodology, we made a series of compounds, but all are basically, both sides are hydrophobic in nature. So it was not actually fitting within these binding sites or active sites. So what we decided to have, either to install a carboxylate, carboxylic acid or carboxylamide derivatives. So we use a basic simple chemistry, ethyl cyanoacetamide or ethyl cyanoacetates, and from a simple benzaldehyde with a multi-step, we were able to prepare carboxylic acid or carboxamide or sulfamide derivatives. Okay, so with these reactions, we are successfully developed around 100 several compounds of this series. And from this series, what we did, we first screen those compounds against purified enzyme in our laboratory. So we first purify the enzyme, check their activity using basic UV visible spectroscopic techniques. We check their activity. We identified around 11 candidates. From there, we again went to cellular studies. We have selected triple negative breast cancer cell MDMB231, which basically express this IDO1 enzyme. In this triple negative breast cancer cell, we saw these 4 compounds are highly potent. These compounds selectively inhibit also the IDO activity, both under in vitro condition and also under cellular conditions. Okay, so we are fine with that. We made the compounds, we check their inhibitory activities, but we have to figure it out, are those compounds actually inhibiting through the active sites or somewhere else? So we again go back and perform UV visible studies, spectroscopy studies. We figure it out, yes, it is interacting with the heme groups. It is, and it is also through the SPR analysis or surface plasmon resonance analysis, we clearly show that these compounds are actually binding to the enzyme, IDO enzyme. It is interacting with the heme group, and it is through circular dichroism study showed that these are not changing the structure of the enzyme. So we are happy with those results, the biophysical analysis. Then we tested our hypothesis, first hypothesis, that the compound should not be toxic. Yes, we are successful. This compound, all this series of compounds we see here, not inducing apoptosis both in normal cells and also in cancerous cells. So we are good with that. Also, we also tested that these compounds are selectively inhibiting the activity of indole 2,3 dioxygenase, not any other enzymes in cellular conditions. Okay, then we thought about, okay, fine, so far we were successful there, but what I said, if the it has to be involved in immunotherapy, then it has to activate the T cells, right? So for that, we collaborated with our Professor Arindam Bhattacharya from Calcutta Universities. So he showed us that, yeah, very exciting results, that these compounds, the selected compounds, 4 compounds, induce the activity of the T cells, okay, under cancer microenvironments and successfully kills the cancer cells. We are not happy with that. If it is like that, we don't know it is really going to work in animal models, right? We can we can test it, their efficacies. Yes, we decided, we again collaborated with them, and we first caused or induced the cancer in Swiss albino mice, and then treated these compounds with the Swiss albino mice through and injected intravenously in alternate days for 5 days. And we saw that after 5 days of tumor implant, 86% of the tumor size were reduced. So we are very successful. We have applied for patent there, and this was published in late 2019. And now, after the successful stories, now I'll be giving few more examples on our approach of structure-based drug designing strategies. So we continued our tosyl hydrogen chemistry. So here what we did basically, this is an indole molecule. So what we try to develop, simple from indole, can you convert to pyrazole? So far it was not reported in this strategy. What we did, we activated the indole either through acylation or the tosylation. Then the tosyl hydrogen involved in 3+2 cycloaddition reactions after opening of the CN bond and can convert in one step to the pyrazole. Okay, so a series of compounds were prepared using this reaction methodology, but we failed. Unfortunately, we did not get very good potency. All the inhibition activities were both under in vitro condition and cellular conditions were in micromolar level. Okay. Then the next example, here what we did, we made an fused heterocyclic compounds. So here again simple benzaldehydes. Simple benzaldehydes. From the simple benzaldehydes, we prepare 4-phenyl 1,4-dihydropyridine, 4-phenyl 2-pyridones, and 4-phenyl 4H pyrene derivative. So what we basically did, we basically changed the electronic properties so that it can interact properly with the heme group, and also you alter the substitution of the phenyl ring so that it can fit properly with the pocket A and pocket B, and we synthesize again around more than 50 compounds and screen it and identified these 3 compounds which are showing nanomolar level inhibition activities in under in vitro conditions against purified enzyme and also under cellular conditions. And now, so we continued our research structure-based drug design approach in several other examples. So now I will focus on mechanism-based drug design approach. I told you guys that you should focus on the reaction mechanism for the conversion of the tryptophan to N-formyl kynurenine. So if we don't remember, so for your refreshment, so you can see that form the superoxyanion intermediate, forms an peroxy transition state, and then it leads to the N-formyl kynurenine. So here, what our strategy is that, can we mimic any of the intermediate or the transition state so that the inhibitors can work, and this approach is called mechanism-based drug designing approach. So here what we did, we made an N-hydroxyamidine-based compounds. A simple N-hydroxyamidine-based compound, if you look at, is actually mimics the peroxo transition states. It was simply synthesized from benzonitrile with an hydroxylamine, hydroxyamidine, and with an NBD chloride. A series of compounds were synthesized, okay, and then tested. Again, we identified these 4 compounds showing potent inhibitory activity both under in vitro conditions and under cellular conditions. And here, in this example, what we try to focus on, another mechanism-based drug designing approach. If you remember, we talked about the peroxo or the superoxyanion basically interacts with the C2C3 double bond that we talked about in the middle of our discussions. Okay, so it forms an peroxo transition state. So basically C2C3 double bond is the main site where the reactions or the catabolism or the oxidation steps takes place. Okay, so can we convert this C2C3 double bond to CN double bond so that we can regulate? And for that purpose, we prepare a several indazole derivatives and screen it, those indazole derivatives, and we saw that these two compounds shows moderate activities, so that our strategy works, but however the efficiency or the potency of those compounds were not good. In next example, again the mechanism based drug designing approach, you remember another hypothesis was there. After the superoxide anion formation of the peroxide transition state, it can form either dioxide intermediate or can form epoxide intermediate. So what we thought, if we make a carbonyl group at the C2 position of the pyrrole ring of the L-tryptophan, we can easily put up further radical addition of the iron superoxide anion superoxide intermediate, okay, so that we can mimic the activity of the indolamine 2,3 dioxygenase. And that exactly what we did. We can convert the tryptophan to oxyindoles, or we treated this isatin derivatives to make these oxyindole derivatives. A series of oxyindole derivatives were again synthesized and tested both on the in vitro and in vivo under cellular conditions, and we identified these three compounds with an moderate activities. So while we were doing research, there are only doing research in India. There are few other groups doing research on the same line, same structure-based drug designing approach, and also mechanism-based drug designing approach in all over the world. And you can see these fields becoming so popular, and every year the number of publications keep on increasing. Okay, and not only the academician like us is working on this field, but also all the big pharma companies, including Bristol Myers, Insights, Newlink Genetics, Pfizer, all are involved in this research field. So everybody think this is the futures. And you can see this epacadostat is considered as one of the blockbuster drugs. Okay, it is few of these are in under clinical trial. Like until 2018, this was in phase 3 clinical trial. Unfortunately, in the middle of the 2018, in July 2018 in particular, the Merck halt the phase 3 clinical trials of epacadostat and Keytruda, the pembrolizumab, the molecular monoclonal antibodies combinations, because it fails the trials in phase 3 trials in skin cancer. Its share markets falls. Everybody, all the scientists related to that field were like upsets. It is a significant setback to this immunotherapy research fields. But scientists came back, and it is hoped that the analysis of the clinical data may lead to an improved dosing strategy or identification of responsive patient subpopulations, and eventually results in one or more clinically successful IDO inhibitor. Because as a scientist, we all know success is not always good. Sometimes failures teach us to learn new things. Okay, and exactly that happens. After 2018 setback, now in 2020, we can now see again 16 phase 3, phase 2 clinical trials are now going on again with the IDO inhibitors, and epacadostat came back. Also indeximod product, NLG and 802 also came back in phase 3 clinical trials, but with a different target, with a different volunteers. So now is still undergoing phase 3 clinical trials. So while this research was going on, we think about, can we do something else? Whatever we have talked about, we discuss two approach: structure-based drug designing and mechanism-based drug designing approach. So this is the strategy. Tryptophan, N-formyl carnitine. This is, if you think about, this is our IDO, and this is the heme group. Can we block the Can we block the conversion tryptophan to this through different mechanism? We got a clue in PNS paper 2018. In late 2018, we got a clue. They reported that the heme group of the IDO is labile. IDO1, the heme group is labile. So the heme comes out and forms an apo-IDO. So this is protein, this is enzyme. So that this protein now is not active, it cannot do it. So heme is required. Okay. So we thought, come up with a strategy. Okay, we thought about, can we make a molecules here which will bind to that apo, not this, will bind to the apo and can inhibit the IDO activities. Okay. So with that in mind, we selected quinine. It's a very popular, one of our ancient drugs from our country, known for Kalazar. So this quinine, and also very popular because this quinine derivatives are used for asymmetric synthesis, particularly in organic research fields. So what we decided to do, we make derivatives of these quinines, okay, and we did it. These are all unpublished works, and what we got it, we identified these three compounds with a less than nanomolar potency, so far not reported in any of the literatures. And these three potent compounds also showing around 2 to 5 nanomolar potency under cellular conditions. We are very excited about that. So when my students came to me with this result, I said, I don't believe it, because this is so far, this with this potency is never reported. So I said that, okay, you do these other things, find out how it is actually inhibiting. Is it inhibiting through the similar mechanism as reported? Then we do permutation combination studies, and what we find out that the inhibitions depends both with the time and also temperature, and that gives the clue. So that means that heme is coming out from the IDO. So in presence of our compound, the heme group comes out from IDO, forms an apo-IDO. But we have to prove our second hypothesis. These compounds are it binding to that apo. So we again did it inhibition analysis. We did it biophysical analysis, theoretical analysis, like in SPR analysis, to prove it. Yes, these compounds are actually binding to the apoprotein. So in inhibiting, it is binding to the apoprotein. So the story is not complete yet. There is a tweak. Another day, my student came to me. He said, sir, there is another tweak is there in the story. What is that? That the molecule, the quinine derivative, whatever we have developed, is showing another activity. Not only it is binding to the apo and assisting the release of the heme group from the IDO enzyme, these compounds, this quinine derivative also binding to the heme group. Because we all know if the heme comes out from the IDO, that free heme group is toxic, because the free heme group can be involved in lipid peroxidations, which can cause a serious toxicity to normal cells or any other cells. And with the several biophysical studies, we prove it. Yes, these compounds are actually can bind to the free heme group also. We checked it, our hypothesis that yes, these compounds, these quinine derivatives, these potent quinine derivatives are not showing any toxicities or milder toxicity to the normal cells and also in the cancer cells. We again collaborated with Professor Arindam Bhattacharya from Calcutta University, and he showed that yes, remarkable result, these compounds actually showing much induced activity of the T cells. So we are hoping that this will be also successful. This will be also successful again under in vivo conditions. So with these notes, I am to conclude here what we discussed so far, that indolamine 2,3 dioxygenase can be an attractive target for cancer immunotherapy, and the search for inhibitors of this IDO1 are going on both in academics and also in pharmaceutical companies. Today I talked about the examples of the synthesis and their efficacies of 1,H indazoles, hydroxyamidine, pyrazole, spiran, oxyindadole, fused heterocyclic 2,H tyrosol scaffold-based inhibitors. Overall, these observations suggest that a simple, you don't need very complicated molecules. A simple heterocyclic compounds can be potent, or even natural products. You can just tweak those natural products which can turn out and potent inhibitor of IDO. So, with this note, I want to mention that in the last 50 years, all the scientists and research community has made a tremendous progress, both in understanding and detection of the cancers. So early detections and application of several strategies can improve the prognosis of cancer patients to an unprecedented levels. I want to show these pictures here. These are the celebrities. Some of you guys know or can identify. The common thing is all are celebrities, but another common thing is here, to me, they are actually the survivor of, some of them are actually the survivor of the cancer patients, the cancers, or they are basically the warrior in cancer. Some of them also succumbs to the cancers. So before concluding, I want to read out one line from a cancer patients. If our children is our future, then why we are not fighting harder to find cures for them? So finally, I like to thank all my students. Some of them are all in doing postdocs in US, Israel. Some of them are doing still doing PhDs research with me. And these are the beautiful, amazing guys. They are very intelligent. So whatever I have talked about is all because of them. Whatever our success, our failure is all because of them. And all credit should goes to these students. Okay. All our research are heavily funded by Dept in particular, and also DST. And also I like to acknowledge, or I like to thank all my collaborators. Dr. Manna, our long-time collaborators from our MSc times. We know each others from NCCS, National Center for Cell Sciences, Pune. Professor Arindam Bhattacharya from Calcutta University for doing our in vivo works, and also Dr. Sachin Kumar from IIT Guwahati. Thank you for your kind attentions. This is our beautiful campus. If anybody is interested, I'll be happy to host you. Thank you.
Thank you, Professor Manna, for such wonderful lecture and introducing us to the wonderful world of immunotherapy, and also sharing some of your very exciting work. So the session is now open for question and answers from the participants. So I'll request my colleague, Mr. Supratim Shui, to please forward the questions he collected to Professor Manna.
Good afternoon. The first question is from Onik Chatterjee.
Yes.
Can high level of hemoglobin more than 16 in blood can cause cancer?
Can high level of hemoglobin cause cancers? Okay, we need to understand things little bit in different ways. So what hemoglobin does? Hemoglobin is also a heme-containing protein, right? Its purpose is to carry oxygens. And this indolamin 2,3 dioxygenase, what I talked about, binds to oxygen. But this is not a carrier of oxygens. And this indolamin 2,3 dioxygenase get expressed particularly in the pathogenic condition. It is not in there in blood circulation systems. So the hemoglobin present in blood circulation systems. So it's two different things. So even if you have a high concentration of hemoglobin, through this approach, we cannot say can cause up cancers.
So the next question is from Sayok. He wants to know what is catabolism?
Okay, good question. So what is catabolism? So catabolism is, think about it's just a chemical reaction. Just a simple, it's a chemical reaction. What is catabolism? What is catalysis? I talked about that indolamin 2,3 dioxygenase in an enzyme. Okay, so what is enzyme? What is catalysis? We need to understand first that. Okay, so let's say A to B. as a chemical transformations. So bond formation or bond reorganization happens A to B, right? From starting material to product. Okay. So this chemical transformation, the rate of the reactions may be slow. Okay. The rate of transformation from A to B is slow. So now if you put a catalyst, so what is catalyst? The catalyst is basically don't directly get involved. It assists the conversion of A to B at a faster rate. Neither it takes parts in A or B, but it assists the conversion of A to B at a faster rate. So that is the role of the catalyst. So when we do actually several organic transformation, we try to use catalyst so that we can get the product in quickly. In cells also we have catalyst. So enzyme is actually called like biocatalyst. So these enzymes basically convert the starting material to product at a faster rate. So here, what I was talking about, the catabolism is basically conversions. Here is the conversion of tryptophan to informal kynurenine. So subject to product, this catalysis process is induced in presence of indolamine 2,3-dioxinase enzyme or the catalyst. It's clear?
Okay, now next question is from Sophik. What is the basis of making an antibody against a particular drug? It should not be antibody.
Okay, I think maybe something else. So the two antibody and drug are the two things we need to clarify. So two different things you need to understand. So what is antibody? Antibody is just like a protein. Okay, so when there is a foreign particle enters in our bodies, our immune systems makes another protein to capture it. Okay, to suppress its activity. It can be a protein, it can be also small peptide. Okay, so these small peptides are the proteins which were actually generated by our immune systems, okay, are called antibodies. And drugs are different things. Drugs are basically which induce or reduce the activity of certain enzymes or certain chemical transformations. Okay, see, we consume water, right? We simply consume water for our survivals. What if we consume alcohol? If we consume alcohol, what will happen? If you consume alcohol in a smaller dose, it may not hamper that much. But if you consume alcohol too much, okay, if you consume an alcohol too much, what will happen? It can cause several other diseases, right? So these higher concentrations of the alcohol can be considered as a drug. Now there are two types of drugs. One is good drug, one is bad drug. So the drugs, I have given an example, some alcohol or other mood-induced drugs. These are called bad drugs. But the drug molecules which are beneficial or which is applicable or which is basically inhibit certain or reduce certain disease conditions are actually good drugs. Basically that can cure those diseases for us. Clear? So these are the two different things. Antibody and drugs, two different things.
The next question is IDO synthesized in our body?
Okay, very good question. Very smart questions. Whoever has asked the questions are very smart questions. You know, as if you remember what I talked about, that indolamine 2,3-dioxygenase or IDO present ubiquitously. That means it is everywhere in our bodies. Okay, particularly what I was trying to say, the mRNA is there all through our bodies. Okay, so the mRNA is there all through our bodies. But when there is an inflammation, When there is an inflammation, when there is a disease conditions, okay, there only, even if you are under stress, there only the due to the inflammations, the cytokine like interferon gamma can induce the production or the synthesis of indolamine 2,3 dioxygenase or IDO enzyme from its mRNA. You know, its mRNA is there. From its mRNA, protein synthesis has to be done. And for that, it needs a signal. And that signal is basically the cytokine or interferon gamma. So whenever those signals are there, then only it will produce the IDO1 in our body. Clear?
Okay, Mr. Sujit Das have two questions. One is, are there any differences in the structure of normal cell and cancer cell? The second one is, how can an anti-cancer drug can distinguish between normal cell and cancer cells to show activity?
Okay, very smart questions. I'll appreciate that questions. Sometimes this kind of questions we generally get it from PhD students, but from BSc students, like bachelor students and MSc student, very good. Sujit, right? Very good question. So, first questions were, can you repeat it? First questions. Sorry for that.
Are there any difference in structure between the normal cell and cancer cell?
Yes. Okay. So, how different are the structure cancer cells and normal cells? It's in a way whenever basically we don't. This is my past experience. It's very difficult to actually explain things. We need to have an eye contact, because then only we can get it. Like whatever I'm talking about, you are trying to understanding or not. But anyway, I'm just guessing that you will try to understand. So, normal cell cancer cells. It's very difficult to distinguish. So that's the problem. That's how I was talking about for the chemotherapy. Most of the patients, cancer patient undergoes chemotherapy, and quote unquote, most of the patients in cancers dies not because of cancer, but because of the side effects due to the chemotherapeutic molecules we induced. So the drug, whatever the doctor puts to those cancer patients, they are actually dangerous, much dangerous than the cancer itself. Because the problem is there, as the question is, there is hardly small difference between cancer cell and normal cells. What are those differences? Few of these I can highlight. Okay. If you want to know more about that, you can communicate or you can send me an email, or you can talk to me later. I'll just highlight few points. The cancer cell and normal cells, chemistry-wise, if I talk about, are different in way. Cancer cells inside pH level is acidic, pH 6.5, outside 7.2. Normal cell, outside 6.8, inside 7.2. Opposite. Cancer cells inside acidic environment, outside normal physiological environments. Second point, cancer cells produce more PD-L1 ligands than the normal cells. If a normal cell produce, let's say, 5 to 10 PD-L1 ligands in its outer surface, it also, cancer cell produce around hundreds. In normal cells, glutathione concentration level is low, but in cancer cell, glutathione concentration is very high. In normal cells, peroxides, singlet oxygens concentrations are low, but in cancer cell, those peroxide and singlet oxygen concentrations are high. Okay, so these are few of the characteristics that how we can differentiate from normal cell to cancer cells, but phenotype and structurally you can completely see they are different. Normal cells have kind of a certain morphology. Cancer cells are very abnormal. They have much more protrusion, the tentacles, because those tentacles are most of the time absent in normal cell. Because these tentacles on cancer cell are very useful because they invade the surrounding normal cells using those tentacles. So from the tentacles also phenotypically, or by seeing Looking at the morphology of the structure, we can identify. What was the second question? Sorry for that. I think you unmuted. Sorry, muted. I think you have to unmute it.
How can an anti-cancer drug distinguish between the normal cell and cancer cell?
Yeah, that is the problem I was talking about. The anti-cancer drugs, whatever we make it, we cannot, simply cannot distinguish between normal cell and cancer cells. It needs a carrier. And that was another our research I was talking about. You know, you have to have a special carrier if you want to administer through the blood. Or if you want to directly inject to the cancer cell, fine. Wherever those cancer cells, if you identify, in particular in the outer surface, skin cancers particularly, if you can figure it out where the cancer cells are, you can directly inject those anti-cancer drugs to the cancer cells. But if it is inside our bodies, you have to deliver. You have to orally deliver it through the blood. It has to go through the blood circulation system. Then you cannot distinguish it. So it needs basically a drug delivery systems which will be useful to selectively carry these anti-cancer drugs to the cancerous cells, not to the normal cells. You know, I'll give an example. Doxorubicin is one of the most popular anti-cancer drugs used for breast cancer in India. This doxorubicin, the formulation is doxil. So the doxil is basically nothing but a liposomal encapsulated doxorubicin. So the doxorubicin is within the liposome of there, the phosphatidylcholine and other, so which can selectively target to the cancerous cells, not to the normal cells. Okay. Any questions?
Thank you, Professor Manna. Due to our, we are running a bit behind the schedule, so we have to conclude this session. So thank you for your wonderful lecture and for the question answer session. So we thank you again.
Okay, thank you so much.
And now our next speaker is Professor Atikur Rahman from Department of Physics, Indian Institute of Science Education and Research, Pune. And before formally inviting him for delivering his lecture, I request Dr. Chandrakanta Bandopadhyay to formally introduce our distinguished speaker. Over to you, sir.
Thank you. Dr. Atikur Rahman is from IISER Pune. He did his BSc and MSc from University of Kolani with solid state physics as the special paper. After that, he went to Saha Institute of Nuclear Physics for his post-MSc and PhD. After doing PhD in the Saha Institute of Nuclear Physics, he has performed postdoctoral fellowship from 2009 to 2012 in Johns Hopkins University, USA, and then from 2012 to 2016 to Brookhaven National Laboratory, USA. And after that, coming from States, he joined the IISER Pune. He has also many awards and credits in his credit, such as he was the Best Student Award from the very childhood. He is a very good student. He has the Best Student Award. He is the Young Scientist Award, and many of the papers that he has published selected as the top 10 discoveries of Brookhaven National Laboratory. In 2018, he got the grand prize at Create the Future Design Contest in USA 2018. And his research interest is in investigating electronic transport properties of nanostructures. Today, he is going to deliver the lecture, the title of which is Nature-Inspired Nanotechnology, Solutions for Energy Harvesting and Self-Cleaning. With these few words, I would request Dr. Atikur Rahman to kindly deliver your lecture. Dr. Rahman, sir.
Thank you for this kind introduction. And yeah, uh, I would like to also thank Principal Maharaj for giving me this opportunity to discuss some about this fascinating world of nanoscience. In the first talk, we already got how nanoscience is trying to solve critical problems of human life. Now, I will discuss here how we use nanoscience to solve other sorts of problems like energy harvesting or self-cleaning. But the major point of my talk will be how we get inspired or how we go back to look at the nature to find the solution of these problems. So first, what I'll do, I will give some examples of existing nanotechnologies that nature uses for long time, who knows, maybe millions or billions of years, they're using it. So I'll just quickly go through some of the examples that we see in nature. So this is my lab here at ISAR Pune. We call it short as a NanoQ because we study both energy harvesting and quantum transport at nanoscale. We have students from all parts of India. Okay, so the first thing the first example that I'd like to take is this lotus effect that is famously known for this lotus leaf. We know that the lotus leaf, it's hydrophobic and it's very difficult to make it dirty, and that is known as the lotus effect. And that's why in various mythology, lotus leaf is considered as a sacred thing. So what it does actually, when you see it from scientist eyes, if you put it under scanning electron microscope, then what you see is that this lotus leaf is actually is made up of some pyramid-like structure which are around 200 nanometer tall. And that is responsible for this hydrophobicity, that water doesn't stick to this leaf. You have seen this thing, we call it water strider. In Bengali, we call it jalpoka. So it can walk on water. And what it has a, this foot is actually made up of some micro hairs. So if you see it under low magnification, where the scale bar is 20 micron, we can see that these are the nano hairs. This feet is actually made up of that. And if you zoom it a little bit more, so now with the 200 nanometer length scale, you see that there are some nano grooves in each of these micro hairs. So each of these micro hairs, there are some nano grooves. And when they put it on top of water, actually these nano grooves, they trap the air, and that helps this water strider to float and walk on water. So there is another interesting thing that is known commonly as structural color. So the beautiful color that you see on butterflies or the peacock's feathers, some birds, pearls, or even some fruits, they are all not really due to any pigment or not really due to any particular color, but that is just due to structure. So there is no actual color present, but the color that you see is due to a particular structure, is the arrangement of those structure in a particular fashion. And actually, Robert Hooke is the first one who noticed this kind of structural color. So if you just take an example of a butterfly wings, and if you say put it under optical microscope, you will see this kind of scale structure. And if you notice that if you touch a butterfly, then there's some dust-like things that comes into your hand, and those things are basically this scale-like structure. Now, if you put those scales under scanning electron microscope and keep zooming, then what you see that they are actually made up of certain particular design. Those are again nearly around 200 nanometer feature size, and they are arranged in a particular fashion. And depending on their spacing or the arrangement, you get all those beautiful colors. But actually, there is no color. Peacock's feather is another interesting example that we can discuss. And actually, Sir Isaac Newton was the first who noticed that this color is not due to pigment, but just due to structure. So if you again look very carefully and if you use say transmission electron microscope or scanning electron microscope, what you see that the peacock feather, they are made of these kind of structures, this rod-like structure, and their spacing and arrangement actually creates this color. Just to show you, like when you see the blue of this part, because some of the part will have blue, some will have other colors. So the blue actually, the color that you see is nothing but the arrangement of some crystalline lattice, these rods of 9 to 12 rods, and they are spaced by 140 nanometers apart. And if you see the green color, that is originated due to the arrangement of nearly 10 rods that are spaced in 150 nanometer apart, and they are arranged in a square lattice. And the brown color that you see, the spacing is around 150 to 185 nanometer. And the yellow comes as a result of these 6 rods, which are arranged at 165 nanometers apart. Let's take another example of pearl. I mean, you spend a lot of money to buy this jewel, but the thing is that it's only calcium carbonate. So basically in tune that we see, and the layers of calcium carbonate is arranged in a particular fashion with a particular spacing, and that gives this nice color of these pearls. And depending on the spacing, the color of the pearl actually varies. So all you can see that around us in nature, this nanostructures are present for a long time. And whether it is due to the color, whether it produces the color of butterflies or this super transparent wing of cicada or glasswing butterfly or this moth's eye. I will discuss this moth's eye later, or the lotus leaf or the gecko foot. So all are as a result of the nanostructures, and that is already present in the nature. Now, what is easy to find in nature is a big challenge for us to mimic in the lab. And the reason that nanotechnology or nanoscience started not too long back is this technological challenges. And now we have overcome some of these challenges, and now we can try to mimic some of these things in the lab. And you will see the complexity to mimic those natural things. So the main challenge is to fabricate these nanostructures with high precision. What you see under a microscope or butterfly wing or moth eye or anything, they look, really perfect. But it's very difficult to make those things in a lab in a large length scale. Also, it's very difficult to kind of make them at a scalable rate, because we can make things in the lab in a small scale, but for industrial application to make it really scalable, it's not that easy. Also, the technology shouldn't be very difficult, because that will add cost in it, because we can use e-beam lithography to make nice structures, but that is not feasible technology for kind of industrial application, because it's too complex and too expensive. And also it has to be kind of, compatible with our existing technology. If we think that we'll replace the entire existing setup with our new things, that is not possible. It has to be initially at least compatible to the existing technology. So today, what I'm going to give you, I'm going to tell you about using a block copolymer. So you chemistry people know much better than me. So how we are using this block copolymer to make these nanostructured patterns, these nano patterns. One great thing about block copolymer is that the processing is a bit simple. It's the technique is scalable, and the technologies what we use is kind of compatible with the existing whatever semiconductor technology. So first I'll give you the basics of block copolymers. You all know it much better than me. So we'll be using here diblock copolymer. It's basically two homopolymer joined together. Here we'll be taking this polystyrene and polymethyl methacrylate. So one block is PS, another block is PMMA. Now, if you spin coat this block copolymer solution, then just spin coated solution, the film is a bit random. But if you anneal them at a high temperature, means not too high, say around 150 degrees or something like that, then what happens is that due to a mechanism called microphage segregation, they form certain arrangement. So what is microphage segregation? Let's take one example of macrophage segregation. It's easy to understand. You take water and oil mixture. That's a classic example. They don't mix. But if you kind of vigorously stir them, and then it seems like that as if they are mixing. But if you leave them for some time, then again they get separated. So this is macrophage segregation because the segregation happens at macroscopic level. Now suppose a polymer chain whose part is like water and other part is like oil. So basically this block copolymer has two immiscible blocks. So one you can think of as water and another part is like oil, but they are attached. They are not, you cannot separate them. So when they tries to separate from each other, so what happens is that the like things, means water-like thing, they will try to come together, all the oil-like thing, they will try to come together. And as a result of this kind of segregation, they likes to kind of, repel each other. I mean, dissimilar blocks, they tries to maximize the, spacing among them. And as a result, they form this kind of nice structure. Now, there is this parameter called chi multiplied by n. I'll just tell you, this parameter is actually important for this formation of this pattern or this microphage segregation. So what are the important parameters? There are three important parameters. One is that the volume fraction of block A and block B. So this how much of block A and block B are present, or what is their ratio? Because I'll show you that depending on the ratio, the final pattern actually matters. And the degree of polymerization, the chain length, basically, if it is too small or too large, then it's very difficult to make this pattern. And there is this parameter chi, which is flow reagent parameter, and that basically tells you the incompatibility. So even if you don't need to understand the details, it basically tells you how much these two blocks are incompatible, how much they dislike each other. So if there is no specific interactions, then generally chi is positive and small. It's not a really large value. So chi multiplied by n, that basically determines the degree of microphase segregation, whether you'll be able to make this pattern or not. That will decide this chi and n. Now, there are different types of block copolymers, and this due to microphage segregation or microphage separation, you can get various kinds of morphologies. Like if you have 50-50 of these two blocks, then what you get is called lamellae or lamellar structure. If you have, say, this is polystyrene, if you have polystyrene 70% and PMMA, this blue is PMMA. So if you have, sorry, it's opposite. If you have polystyrene, which is say 30% and PMMA 70%, then you get this kind of structure. If you have the other thing where the PMMA is 30% and polystyrene is 70%, then you get this inverse cylinder, or the other ratio will give you the cylinder morphology. So the way I will designate them from now, I'll call them MXX, where M will tell whether it is cylindrical or lamellar or it's inverse cylinder. And this XX will basically tell you the molecular weight in kilodalton. So if I say something that is called C99, that's basically the pattern is cylindrical pattern and the molecular weight is 99 kilodalton. Now, when we try to make this a structure, this substrate that plays a very important role. And most of the failures of making this pattern, apparently simple pattern, is due to this substrate. The reason is that these two blocks, this blue or red block, they should be neutral to the substrate. Means their interaction to the substrate should be kind of similar. But if the substrate is non-neutral, that means that one of this block is preferred to the substrate, then what you will get is kind of out of plane stacking structure for lamellae, or if it is cylindrical, then in plane lying cylinder for the cylindrical structure. But there are ways to make this substrate neutral. So what we generally do, we coat a neutral layer on this substrate, and basically make the substrate neutral, and then we make these structures on top of that. And now you can get your desired lamellar or cylindrical pattern on top of this substrate. But there is a problem of using this pattern as it is, because these are soft material, these are polymers, so they are not compatible to most of this, semiconducting processing or other processing, which we have to expose them in harsh environment. So most of the time, what we do, we convert this pattern into some inorganic pattern. So how we do that? Let's take a piece of silicon, and I want to generate pattern on the silicon. So I'll spin coat this polystyrene PMMA block polymer on top of it. So as spin coated pattern, if you see, it's there is no oil ordered. You cannot really see those patterns here. But if you anneal them, then now you can see the microphase segregation. So basically, we annealed it at 200 Celsius. And now you can see these dark places are basically the PMMA regions, these PMMA regions, and these continuous places are the polystyrene part. So basically, for imaging, we remove those PMMA to show the image clearly. But to convert this PMMA part into some inorganic thing, what we do, we take the advantage of a technique that is called sequential infiltration synthesis, where we preferentially load this aluminum oxide on these PMMA regions. And this is done with a machine called atomic layer deposition. It works in exposure mode. So you preferentially now load aluminum oxide in these dots. So we still have this polymer matrix present here. In the next step, what you do, you burn this polymer matrix using oxygen plasma. So you remove all the polymers, now your substrate, and you left it with these inorganic aluminum oxide dots on top of this. So this is a piece of silicon where these aluminum dots are present at all. And you can see the length scale, this is 100 nanometers here. And the best thing about is that it's highly scalable. So you can see that you get these kind of pattern over a large area, and they're pretty nice. And though the technique is kind of simple, but you can generate this small few tens of nanometer nanostructure over centimeter, or even a kind of anything where you can spin coat this. So it's pretty scalable. The best thing about this block of polymer is that just by tuning the molecular weight, you can change the size of these dots or the spacing between these dots. So it's pretty tunable. Also, you can control the how much material you want to load. So here you can keep the spacing same, and you can make the dot size bigger and bigger by loading more and more amount of material. So this is another kind of degree of advantage for using this technique. We also came up with a technique to use this block of polymer to make a multi-layered structure. So basically making three-dimensional nanostructure. This is a bit involved thing, so I'll not go into details of that. But thing is that we can make kind of any kind of structure on top of another. So if I just take two examples here, so this is a lamellar structure, and this is a cylindrical dot pattern here, and we can make the combination of these two, and you can see those dots are sitting on this lamellae pattern, and you can see the structure is observed over the entire substrate. Another good example is inverse cylinder and cylinder. So here, this is basically the whole structure. There, these are the holes, so porous membrane kind of thing, which is made with this kind of block of polymer. And this is for the cylindrical. Now, if we combine them, if we try to make a top of another, then what you get, you can place these dots just near to these holes here. And in a low magnification image, you can see it here, it's observed over the entire substrate. And we explore almost a huge diverse of morphologies using this, and all of them are kind of non-native to block copolymers, because if you remember the block copolymer structures, you can get only basically three different kind of structure, primarily three. I mean, there are some other complicated structures available, like lamellae, cylindrical, and inverse cylinder. But in this case, you can make kind of various other types of structures that you can think of. We did some standard characterization for that. So here, basically, we made only two structures on top of another. So if you put like C on top of another C, you'll get this kind of structure. Then cylinder on top of maybe lamellae, you get this kind of structure. So there is a whole zoo of structures that you can get. You can cross-check them with synchrotron data to see the structure, whether it is over a macroscopic scale or not. And here we made like three different structure. We took lamellae, all are lamellae, but what we see that these lamellae, they basically anti-aligns each other when you put another lamellae on top of it. So it basically makes this net-like structure. You can make even a complex structure. So in this bottom one, we made first this lamellae, then again a different molecular weight lamellae, again a different molecular weight of lamellae, and then put cylindrical structure on top of it. So you can see those structures. And they're pretty small. As you can see that the length scale is only 50 nanometer. So now, okay, this is fine. I mean, we have the control on how we can grow these nanostructures, how we can make these patterns. So what are their use? So first I will discuss about this, their anti-reflection properties, how you can use this nanostructure to solve some problem. So what is the problem? I mean, we probably all come up with this thing that when we call someone which is far apart, we generally mock up our hands to, shout so that our sound goes. Or if you know this musical instrument, they have this kind of structure. The reason is that the problem that we want to solve by doing this is the impedance mismatching. What is the problem? If wave traveling from one medium to another, it gets reflected due to the mismatch of your impedance. When you try to shout larger, your area in your mouth is small and the outer space is big. So there is a mismatch of this impedance. So you try to put your hands near your mouth and try to make something funnel kind of shape so that the impedance gets matched. And this impedance mismatching problem is observed in various fields, starting from electromagnetism to acoustics, seismology, almost everywhere. So how you try to solve this problem? Let's consider this reflection problem. So light, when it falls from one medium to another, it gets reflected. Say if we take a small, say, piece of silicon, which has an average refractive index of 4. So when light falls from air to silicon, this refractive index abruptly changes from 1 to 4. So that's a huge change of refractive index. As a result, on an average, 35% of the light gets reflected from silicon. The same thing happens with glass. That's why you see a bluish coating on top of it, because what it does that this transparent dielectric coating, actually it tries to minimize the reflection, but that is effective for a particular wavelength because it relies on the destructive interference. So most of the coating that you see on your camera lens and other things, they are optimized for a particular wavelength. But we can explore another technique where this reflection we can cut down greatly, and which can be of useful for various wavelengths over a broad spectrum. So what we do, if we make this kind of nano cone structure on top of this silicon, and if this spacing between this nano cone, they are deep sub-wavelength, means they are much smaller than the wavelength of light, then what will happen is that if you now just take slice from this top, then what will happen is each slice will contain part of the silicon and part air. And as you go deep inside, the volume fraction of silicon will keep increasing. And if this structure spacing is much smaller than the wavelength of light, then it will not see it as a individual element. What it will see as an effective medium whose refractive index is gradually increasing because the volume fraction of silicon is gradually increasing. So now you don't have this abrupt change of refractive index as you go deep inside this structure. Rather, it matches really nicely or smoothly from this refractive index 1 to 4. And as a result, this light will get going inside this structure, and this is independent of wavelength. So it works over a large wavelength range because there is no restrictions on a particular wavelength. So it has a broadband characteristics. And this actually is, people got motivated from this moth eye geometry. This is the moth eye. I mean, if you look it under scanning electron microscope, there are nearly 200 nanometer tall, this pyramid-like structure on this moth eye. As a result, what happens is that during nighttime, whatever the little bit of light that reaches to their eyes, so little bit of photon that comes in, they can absorb those photons and they can see, but due to the lack of reflection from their eyes, their predator cannot see them. So that basically helps to moth, I mean, like helps them to be alive. So now we try to basically mimic this moth eye structure in the laboratory. And it's a bit, I mean, difficult, but with using this block copolymer technique, we can do this thing. And so what we did, we made this kind of moth eye structure on silicon, and this is in around this 200 nanometer length scale. So just to give you a feeling of this length scale, if you consider this, all these are human being, then with the 10 to the power 10 or 10 to the power 11 number of these human being on, say, on this small area, what will happen is that the whole Earth's population, which is around 8 billion, you can accommodate them on a surface of, say, 1 rupee coin. That is sufficient enough to accommodate all these human beings on this Earth, because that's only 10 to the power, say, less than 10, I guess. It's 8, 10 to the power 9. That's the total population. So it's really tiny. So what we do, we start with a aluminum dot decorated silicon substrate, and then we use a technique called reactive ion etching. What it does, it basically preferentially removes the silicon anisotropically, and these aluminum oxide dots that serves as a hard mask, so it doesn't allow to this substrate get etched underneath, but this open part, they gets etched. And as a result, as you etch, say within 90 seconds, you can get this kind of nice structure over the entire substrate. And again, you can tune the pitch by tuning the molecular of the block copolymer. So you can make like 30 nanometer pitch or even 70 nanometer or anything in between. You can also use or tune the RIE condition to make this highly spiky kind of structure, or like any kind of structure that you want. So you just have to play a little bit with this RIE condition, reactive ion etching condition, to get the desired structure that you want. So what we found is that as you keep etching, so we make this structure taller and taller, this surface becomes less and less reflective. And around a 200 to 25 nanometer height of this structure, we see that the reflection gets reduced by 99% over this entire wavelength range. So it has this anti-reflection characteristics, and it works over a large wavelength range. And another interesting point is that what we found that even at large angle of incidence, the reflection is less. So even if you go to even 60-degree angle of incidence, we see less than 5% of the lights gets reflected. And actually this is very important for designing solar cell, because if you see that sun doesn't always shine on top of our head, so for making or increasing the efficiency of solar cell, there is something called sun tracker, where the solar cell actually keeps moving. It tracks the position of the sun to maximize the light, maximize the absorption of light. But if you can make this kind of structure atop of this solar cell, then you really don't need this sun tracker, because even when the sun goes near the horizon, it still can absorb a lot of light. The reflection will still be very low. So to test our this efficiency of nanotextured anti-reflection thing, what we did, we just took the solar cell silicon and made a solar cell. So we really didn't take care of any of the specification or any fine cleanliness. So whatever the solar cell that we made, we even made the PN junction by ourselves. So we got 8.7% efficiency. But then when we made this textured on top of that, the efficiency increased to 13.1. So there is a 50% enhancement of efficiency. And what we found that the efficiency is increasing only due to the increase in the short circuit current. So this is called the JSC, or the short circuit current, and there is no change of this VOC here. So what it does, I mean, if you don't know anything about solar cell, the increase in the short circuit current, the photo current, means that whatever the photon is going inside, it gets converted into this electron hole pair, and you can get current out of it. So if you can increase the absorption of light, you get more and more current out of it. And we do see that this 50% enhancement of efficiency is exactly matching with the enhancement of this photocurrent. So basically, whatever the more amount of light is going inside, that is getting converted into current. And that helps to increase the efficiency of solar cell. We did some fun stuff using this kind of nanotexture. You know that glass, if you take a piece of glass, it generally reflects around 8% of the light from, basically 4% from each surface. So total around 7 to 8% from both the surface. So we took a small gas slide, then we made this nanostructure on both sides of this glass. So here, if you see very carefully, there is actually a glass slide, which this piece, circular glass slide, which you cannot see. Probably you can see this corner at one end. The difference between these two slides is that in this glass slide, we didn't make anything. So there's a flat substrate, but in this, we made this kind of structure on both sides. So it's cutting down the reflection from the top surface. Also, it's cutting down the reflection from the bottom surface. Because when the light goes out from this glass slide to the outer environment, this nanotexture is also helping to match the impedance so that light can transmit easily. So this is a flat piece of glass. As you can see that the reflection, I mean, the transmission is not really that high in compared to this nanotexture, double-sided nanotextured sample. So it's basically kind of super transmitting. And we found that the reflection is reduced by 96%. And actually this has a lot of use in high power laser, where you can make the optical equipment which has a higher laser damage threshold. And we do found that this double-sided textured glass substrate has a high damage threshold. Now, during the introduction, at the beginning, Principal Maharaj was saying about the solar cell getting dirty with this dust. Yes, that's a big problem. And it's probably less problem for us, but where you get a lot of sunlight, especially the desert area, you expect that solar cell will kind of revolutionize, but the big problem is the dust or accumulation of this sand on top of this solar cell. Now, what we found that this conical structure, they are actually very good for this hydrophobicity. And anything that has hydrophobic property will not allow their top surface to get, the dust to accumulate, or even if it rains, it will easily take out those dirt. So just to give you some introduction, so if you put a water droplet on a substrate, and if it makes a meniscus like this, so if this angle is less than 90 degrees, then the substrate is known as hydrophilic. But if it is more than 90 degrees, call it hydrophobic. And once it is more than say 155 degrees this angle, then we call it super hydrophobic. And this is just a comparison. This is the ACM structure that I discussed earlier as a lotus leaf. So it has this nano cone kind of structure. And this is the structure that we made on top of silicon to test the super hydrophobicity of this structure. And yes, we do found that if we make a hydrophobic flat silicon, the contact angle is 110 degrees. But if we make this kind of spike structure, then the contact angle increases and it becomes more than 160 degrees. And what we found that it can even withstand to the highest speed of raindrop, which is 20 miles per hour. And we can see one just quick video here. So if we make this kind of structure, then they really repel this water from this nanotextured substrate. It gets splashed on top of this. This is again, and it can withstand the 20 miles per hour, the highest speed of the raindrop it can withstand. We also use this structure to make something that is called anti-fogging. That means fog will not accumulate on it, especially if you're driving on a rainy day or maybe in a, yeah, it's generally is very much visible on that. So what happens is that when these water droplets accumulate on a flat substrate, they make a bigger droplet. But if it's a nanotexture, then when they accumulate, if it makes a bigger droplet, then it jumps out of it. It's called the jumping droplet phenomena, and that happens due to this large droplet of this thing. Because when you make this large droplet, this asymmetry is broken and the addition to the substrate is actually reduced. So what I'll do, I'll show you a small video where this left side, we made this kind of post pillar kind of structure, which will show some anti-fogging, but not that much. But this highly pointed structure will show really efficient anti-fogging ability. So here you can see that it makes this bigger droplet, but when these two droplets are coalescing, they are jumping out of this surface, thereby keeping the surface clean. So it's very efficient for anti-fogging application as well. So with this, I come to the end of my talk. So these are the people whom I should acknowledge, with whom this work has been done. And thank you all for your patience. And now I'm ready to take question.
Thank you, Rahman, for your wonderful lecture and introducing us to the world of energy harvesting using nanotechnology. And now the session is open for question and answer. And I would request my colleague Supratim to please forward the questions he has collected to Dr. Rahman.
Hello. Am I audible?
Yes.
Okay, the first question is, how a particular structure depicts a color? What is the mechanism?
Okay, so again, a particular structure is due to the interference. So the difference between the pigmentation and structural color is that in pigment, like say if you consider turmeric, okay, it absorbs the light. It absorbs the light. So the light basically excites the electron and it takes it to a different excited state. So light gets absorbed on that. But in structural color, that color appears due to the interference. So whatever that is coming out is others are kind of forming the destructive interference, and whatever is existing, the space, depending on the spacing, you see that particular color. So it's not due to absorption.
Okay. The second question is that what are the limiting parameters of harvesting sunlight for the use in running vehicles?
Okay, I think the main is the energy density, which I understand for running vehicle. If it is a low power vehicle, then probably it's easy to run a vehicle. I mean, we already have some vehicle that runs in sunlight, but it's mostly due to the energy density that you need. So generally we combine with some other mechanism. Even that's why the concept of hybrid is coming, even when you're trying to make this electric vehicle. So you're trying to get the quick power from maybe a supercapacitor, a discharging of supercapacitor or something, and then try to run it with a constant, consumption of power, which is not that much. But what you need is more and more efficient solar cell to overcome these problems. That's it. And it should be kind of flexible in one sense, that it should be rugged to be really useful for vehicle. Area should be small. So the efficiency, maybe multi-junction solar cell can actually help, because there are certain thermodynamic limit where a silicon solar cell cannot be, 100% efficient because of thermodynamics. So it can go maximum up to certain, I think 35%, I think the upper limit of this efficiency. But if you use a multi-junction solar cell, then probably that is possible. And that's how people are trying to overcome this problem.
I think there is no more relevant questions. Thank you very much, sir.
Okay, thank you. Thanks a lot for inviting me. Yeah, okay, thanks a lot.
Thank you, Dr. Rahman, once again for your wonderful lecture, and we hope to listen to you in some later time also. So thank you.
Thank you.
And our last speaker today is Dr. Niladri Patra, and it is a very proud moment for all of us here in the Department of Chemistry to have him as a speaker today. So before inviting him, I think I would request our Dr. Chandrakanta Bandyopadhyay, head of the department, to introduce his student, Niladri, formally. Over to you, sir.
Thank you. Now it's a very nice time for me because I'm going to introduce such a person who was at one time he was my student, and really I am very proud and feeling very happy today. As I know him from his undergraduate time, so I must tell a few more words about him. From the very beginning, I saw him very much energetic and enthusiastic. From the very beginning, I watched it. And now at this age, when I came at this age, at the end of my life, almost, then I see that Swamiji told that the whole world has been made by the energy of man, by the power of enthusiasm. And really, these two ornaments, the energy and enthusiasm, both ornaments are in his character. As a teacher, I wish him success in his life. Okay, now I am coming to our official job. That is, after doing his BSc from our college, he went to IIT Delhi for his MSc, and then he went abroad for his PhD work, and he did his PhD from University of Illinois, Chicago University of Illinois, under Professor Krell, and he did 2 postdocs, of which one is at MIT. You know, it's a very prestigious institution. He did postdoc there from 2014 to 16, and then he went to Brookhaven National Laboratory. I think there he met Dr. Rahman there, because they were almost at the same time at that institution. And after that, he came back to India and joined the IIT Dhanbad, which was earlier known as Indian School of Mines. And he has also many awards and fellowships in his credit. I'm not going into all. Some of them are Amra Fellowship, some Prabha Devi Memorial Prize, and etc. Whatever it may be, I am not going into these details. His topic today is multi-scale modeling of self-assembly and nanostructures and nanomedicines. With these few words, I request Dr. Patra to kindly deliver this lecture. Dr. Patra, please.
Is that okay? So thank you very much, sir, for kind and introductions, and I would like to thank respected moderators and other professors, and good afternoon to all students. I think it's visible, right? Is that visible? I'm audible?
Yes, yes, you are audible and so visible.
Okay, so
Niladri,
that's
please
great.
continue.
Okay, so today I'll be talking about the multi-scale modeling of self-assembly processes. And today we'll be talking about 2 processes mainly: how we can build a bigger nanostructure from a building block, and how we can use a theoretical tools to understand the nanomedicine process. Okay, so here are some graphical pictures. Okay, by the way, this is a complete theoretical work. There is no experimental involved unless I'm told. I'm going to tell you. So the first part of that talk today, I'll be talking about the self-assembly of the graphene nanoribbon. How we can guide the nanoribbon to make a particular structure, nanostructure, and then we'll talk about how we can make a kinetically micelles, which can be used for a drug delivery. And then we'll talk about another micelles process, which is also experimentally verified, that certain structure is particularly necessary if you want to deliver a certain kind of drugs. It depends on this the ratio of the hydrophobicity and hydrophilicity. And then we'll talk about enzyme catalysis. And this enzyme catalysis is actually probably all you know. It's related to the Parkinson disease. We'll talk about more in details later. And then finally, we'll talk about the drug resistance problem in our body and who is responsible for it and how we can deal with it. So as I was told that most of the undergraduate and MSc students probably do not know about theoretical chemistry and computational chemistry. In this talk, I focus mostly some particular method called molecular dynamic simulations, which is very simple idea. We just wanted to see what is the atoms are, what are the atoms are doing if you put all together in computers. Okay, so this is called the computer experiment or the simulations. And for that, it's very simple. So molecular dynamic simulations is a computer simulation technique that allows one to predict the time evolution. This is the important word, the time evolution of the system of interacting particles. So basically, we put all system together. The system can be consist of atom, molecules, organelles, anything, and then we'll run the simulation. We'll see what is the time evolutions of the interacting particles. So for this, we just need two kind of stuff. One is the initial conditions. Okay, so what are the initial conditions? The initial conditions is initial positions. Let's say if you wanted to simulate a protein, for the protein, the initial position can be taken from crystal structure. I hope everybody knows this NMR or some other X-ray crystal. Okay, so you can always get the crystal structure of a protein from a crystal structure database. Okay, and the velocity of all the particles in the system. We'll talk about the velocity, how you can give an initial velocity of a system. So this is the first criteria. You have to give the initial conditions to the systems. Okay, and the second part, which is the interactions part, potential. We need to tell computer how are these atoms are going to interact. What is the mathematical equations? What is the mathematical equations, how they know each other. So that is basically called the potential. For example, if the two particles are interacting only using this van der Waals equation, then this Lennard-Jones equation is your interacting potential. If there is nothing else, then this is just a simple R6, R12. This is also called 612 potentials, nothing else. Okay. And the time evolutions, will be calculating the time evolutions by just simple Newton equations. You can see this. This is after nothing but as Newton equations, and these are the later derivative. We just replace these accelerations by velocity, and then velocity is by the positions. Okay, so if you see that in this case, if you want to solve this, you need a force, and you also know that there is a relation between this potential and the force, which is basically a gradient, nothing but this one. You see, so what you need in order to start a simulations, computer simulations, you need the initial positions as well as velocity, and also you need this potential U. Okay, so once you have all these three kinds stuff, then you can start a simulation, and then you wait for this time evolutions, and then you see what is happening inside the system. And if you remember your classical mechanics from your BSc class or MSc class, if you know about R, which is the position of the system, and the momentum, okay, but think about the Newton mechanics. If you know the R and the momentum, in principle, by using these two properties, you can calculate any physical property of the system. I'm not talking about quantum here, although you can use any quantum property by using these two properties, but in a quantum world, they are differently called. They are called operators, okay? They are called the momentum operator and the position operator. But in the classical mechanics, they are called just simply momentum and the position. So if you know the momentum and the position, you can calculate anything, any physical thing from your system. Okay, that's the goal of the computer simulation. So that means when you are calculating this R and T, we are making a movie over the time to just register somewhere what is the position and what is the momentum of each individual particle. So once we have it, we are done. Okay, so now what is the advantage of this computer simulations? Why not do always experiment? Okay, But think about some certain cases, you cannot really do experiment. For example, a protein, what is happening inside a protein? You cannot do really all experiment because it is very delicate to do that. We do not have that kind of sophisticated technology yet. But if you use this kind of computational tools, you can see exactly what is going on inside it. We'll talk about later. Okay, so these are kind of advantage that you cannot do at all experiments. Then you really need to have this kind of computational tools. Okay, so this is one of the famous potential that we are talking about. Remember this A, you need this U. Okay, so this is one of the well-known and verified potential which works very good. Okay, so this is like a bond to energy part, this is an angle part, this is a dihedral part, impo part, probably do not know what is impo part. It's kind of dihedral, but it's out of the plane. Okay, and this is the van der Waals and this is the electrostatic. So in that means in our system, these are the compositions of your potential. Okay, for example, if there is nothing, if the two particles are only interacting with this vertical, this van der Waals, then only this U equal to these van der Waals parts. I hope you recognize this part. And this is the electrostatic part. If the two particles are charged, then the equations which governs this potential is just nothing but Coulombic interactions. Okay, so you see these are the different components of the potential, and then you need to solve this equation to you need to basically get the gradient in order to get the force. And once I have the force, I apply force on each particle, and the particle will move. Okay, the particle can be a bigger molecule, the particle can be atom. We'll discuss later. Most of the cases, in this case, it is atom. So I calculate what is the acting potential on each atom, then I calculate the gradient because I know what is m of that particular atom. Then I get the force. Then I apply the force on the atom will move. The next step, okay, again I do the same, then the atom will move. Now think about it, I have 100 atoms in my system and I do all kind of stuff. Then every time step or every cycle you'll have a new configurations. Okay, so you'll have a new configurations where all atom will move for a particular cycle. So for cycle number 1, 100 atoms will move. Next cycle, all 100 atoms will move to the second position. So the first position is a configuration 1, and the second position, second cycle, have a different configuration of the system. It's called configuration 2. So now you have a millions of configurations. Then what will happen? You will have a running movie from configuration 1 to 10 million configurations. Then you run a movie. You will see a sequential movie how the system evolved over the time. This is the simple idea about molecular dynamics. So now I will show you some of the applications of the molecular dynamics simulations, what we learned so far. Okay, this is computational, this is too complicated, but this is very simple. You define initial position and velocity. By the way, how do you define the velocity? If you remember the Maxwell-Boltzmann's law in a gas, so you just need to give the temperature. And if you remember that law, from that, if you define the temperature, the random velocity on each atom will be automatically defined. Okay, this is just a backtrack. So you give the temperature, and the temperature basically in the vice versa, it will use the Maxwell-Boltzmann law to get the velocity on each atom. Okay, and then you calculate the force. Then you move the atom, you get the movie. When you get the movie, you get the positions and you get the momentum. Once you get the momentum and the position, you can calculate any property of your system that you want. Okay, that's the simple thing. Okay, so now here are the applications. So what you did here, this is a simple graphene sheet. Okay, so initially what we did in a simple relaxed graphene sheet, we just put a single water droplet. Remember, this is a 2 droplets from upside and downside. So initially we put 1 droplet and we see actually there is a dent. Let me do something a little different. So remember, this is my graphene sheet. Okay, I'm not so, and you put a 1 droplet like this. Okay, and then you try to simulate. After some, what will happen? You see, the last figure is also talking about this nano surface. This is a nanoscale material. Okay, this graphene is also nanoscale material. So what will happen? Because the water is hydrophilic and this one is hydrophobic, so it's the contact. It doesn't like too much. So what will happen? It does not like too much, but it's also not so bad because it has some charge. So it will be little bit dispersed somewhere, something like this. So basically, it's like a something like this. But not only that, what will happen? It will make a curve on the graphene. something like this. Okay, this is the dent, you see, because it is not so easy to deform the droplet. So basically, there is a negotiation between the deformation energy of the graphene sheet and the deformation of the spherical shape of the droplet. Okay, so, but if you put two droplets on the top of each other, then what will happen? They actually not sitting on the top of each other. You see, they're slightly shifting, and this is because they are very smart. Look at this. This guy created this dent, this dent, and this guy is actually enjoying this dent. It is not actually using its own energy to create a new dent. It's basically they're helping each other. So this is the equilibrium state. Okay, but if the graphene layer is fixed, then they will sit together exactly on the top of each other. But if the graphene is not relaxed, if the graphene is relaxed, they'll never sit on the top of each other. They will always go by this way and this way because they want to enjoy this kind of curvature. Okay. So given the fact, we know that water droplet actually can induce a dent on the graphene sheet. So we devise a new thing. Okay, look at this. So what we did look at this lower panel. Look at this lower panel here. So this is like a star shape. There is a particular reason why we make it a star shape. We'll discuss a little later. We put a small droplet and then we start to simulate. And you see actually, they immediately start to close. This is like a flower. Okay. And you can see that if this graphene wants to close, or if this flower wants to close, it has to pay a negative, this positive energy. Okay, the positive energy is kind of bad energy in a sense. Okay, that means the graphene has to bend. This graphene has to bend. So we actually help this graphene by making this narrow thin strip. If the strip is completely like this wide, then it has to pay more energy in order to bend. But we just help them by just making this kind of narrow thin. Okay, so that's why the bending energy for the graphene is less, but it is now basically making this flower because of this interaction between the water and the graphene. And this is purely van der Waals because the graphene layer is neutral. In this case, actually look at this. So you put a droplet here, it first forms this one like this, and then it basically spits out the water. Okay, that means water is actually the graphene does not like the water, but actually graphene is using the water as a catalysis. In order to make this structure from A to B, the graphene has to bend, but there is an energy to pay for this bending. Think about the Arrhenius activation energy plot. There is activation energy. And this activation energy, in a sense, in this case, the bad energy is the bending energy. So you have to overcome the bending energy. And in order to overcome the bending energy, the graphene is helping. The graphene is taking the help of the water. And that energy is graphene water energy. This graphene water energy is helping to overcome the bending energy of the graphene. But once the graphene see each other, then the van der Waals energy between graphene and graphene is much higher compared to van der Waals between graphene and water, because water is hydrophilic. It does not really like it. So that's why, in order to enjoy more energy, the graphene basically what it does, it splits the water and it now goes in as only graphene layer. Think about this one. Okay, so what we did now, this is a long nanoribbon, and we put a small nano droplet. Okay, and after some time, you see this kind of stuff. We'll show you a movie, then we'll understand what is the difference. Okay, and then we understand that maybe if we put a bigger droplet, we can see different structure. Okay, see, look at the difference. If you put a smaller droplet with a similar size of nanoribbon, I'm talking about the width, not about the length. Length has nothing to do. It is about the width of the nanoribbon, which is important in this case. If you put a smaller droplet, you see this kind of structure. But if you put a bigger droplet, you this rolling structure. So we call this one is a sliding structure, and we call this one is a rolling structure. Okay, and now look at this, what is happening. If you have a bigger droplet on the nanoribbon, the tip of the nanoribbon is always covered by the water droplet because the water droplet is much bigger. So here the total energy between the graphene and the water is much higher compared to the bending energy here. So the bending energy of the graphene is lower than the graphene water energy. But in this case, look at this, what is happening when it is, see the tip is not attaching to this water, because the water is very tiny here. It doesn't have enough water to hold it. Now here is basically hesitate. Should I roll or slide? Should I roll or slide? But now there is a negotiation between the bending energy and the water graphene energy. The bending energy is much higher when this guy is here. Look at this. This is the understanding. This is the principle that we need to understand. So in this case, this bending energy is not overcome by the graphene water energy. So then it slides. So when it slides, it is happy because it sees the graphene van der Waals. Okay, so that's how you just tune the water size, and you get a different nanostructure. So this is also self-assembly. This is also called the guiding self-assembly. Okay, and which is important in actually, if you see the previous speaker about the nanotechnology, he was saying that nature has amazing features that most of the thing actually made by self-assembly. So the self-assembly has an advantage. You do not have to pay more energy, okay, because it's self-driven. That is the advantage. It's a bottom-up approach. But if the top-down approach, you have to pay huge amount of energy in order to make a particular structure. That is the essence of the bottom-up approach. You do not have to pay a lot of energy. You just guide the structure. Once it is there, it will be there forever. Okay, so now what you understand that maybe we can have a different structure if you create more complexity. Think about this structure. Here, you can only introduce the chirality of the nanoribbon. I hope you know all chirality of the graphene on nanotube. Okay, so if you so here we cannot introduce the chirality on the droplet, but we can make it more complex system by introducing the chirality on the nanoribbon as well as the nanotube. So look at this. If you place this nanoribbon on perpendicularly on the nanotube, you see this rolling structure. But if you put slightly like an angle, then you see as a rolling structure, but it's some kind of, like a DNA type structure. Okay. So actually, there are all mathematical models. The mathematical models I didn't put it because it is too boring to discuss the mathematics. Okay, look at this, how it works. But here is important thing to understand. So this has nothing to do. You just put it perpendicularly, it will make a roll structure. They are happy because they're enjoying the graphene van der Waals energy on the top of each other. But the question is why it is going on the top of each other. The rolling has a multi-layer. Think about it. As long after the first layer, the radius of the ring is increasing. You go back to this. It will be much easier to explain. Think about this one. So once I have the first layer, the second layer has a larger radius compared to the first radius. The larger radius means you have to pay little bending energy. So this is favorable. Okay. But if it goes by this one, you see the radius, the pitch rate, the radius of the ring, of the graphene ring, the nanoribbon ring is same. So the bending energy is same, but it has another advantage. It is called the side-side interactions. Okay, these side-side interactions, which enjoy the side-side interactions. So these are the two different mechanisms by which it basically forms a nanostructure. We can similarly put a graphene nanoribbon inside the nanotube. Okay, and it can form like these structures. Okay. But this is all like a room temperature, but there is these are kinetical structure. I will explain why it is kinetically not the thermodynamic. Okay. So actually then later they found experimentally the similar kind of structure in experiment, which is kind of nice to have it. Okay. But why I say this is a kinetical structure, not the thermodynamic structure. Look at this simulation. So what happens? It starts from this side, it goes all the way because of the momentum, and then it comes back. Okay, then it comes back and inside make it liquid. The advantage of this graphene nanoribbon or the yellow one to be inside, it can enjoy the whole van der Waals between the nanoribbon and the nanotube. Okay, this is a kinetical trap. This is a meta structure. But thermodynamically, this is not favorable. What means if you really place this nanoribbon very slow inside this limiting distance nanotube, it will never roll. Okay, so you see, it has to pay the bending energy, and this bending energy coming from the van der Waals. But then there is another factor which is called the temperature, this KT. The KT is also helping. That is the kinetic factor, which plays the role to make this DNA type on the structure. But if it is a, let's say, 10 Kelvin structure, it will never roll. It will go like this. So if it is a very long nanotube, and if you put a smaller nano ribbon, it will never roll. It will never roll, because that is bad, because it has to fold it. Okay, and the fold means you have to pay the bending energy. Okay, that's bad. Okay, so that's over. So we have these mathematical models and all the equations, and I intentionally skipped it because it is kind of complicated and for the undergraduate students, but qualitatively you can understand is what is happening. Okay, so now the next thing what we wanted to introduce, it's a different kind of simulations. It is called the coarse grained simulations. Okay, so when the system is very big, for example, I don't know, 100 millions or 200 million atom, then you cannot really consider a single atom as a single particle. We do not have that big computer. Okay, so we do not have the big computer. Even the fastest supercomputer in the world cannot simulate that. So what we need to do is, maybe we can consider certain group of atoms as a single particle. Okay, so then these are called bits. So in this case, actually, we did the same. We use a technique that we do not consider all atom as a single particle. Rather, for example, this is a benzene. So benzene is not considered as a six atoms. So now this is actually 3 particles or 3 bits. This is coarse grained model. Okay, this is the difference. Okay, so what is the origin of this project? Okay, so this is actually related to the drug delivery vehicles. Okay, imagine you want to deliver a drug inside your body using micelles, which is actually very standard now these days. Okay, micelles is largely used for the drug delivery. But if you want to make a micelle which is thermodynamically stable, then there may be the less number of drug inside the micelle, which is called payload. Okay, so the number of drug molecules inside a micelle is called payload. Okay, but can we make a kinetic micelles which has a drug much higher payload and sufficient stability so that it can deliver the drug from the point of injection to the cell? That is the goal. So can you make a kinetically stable micelle? Okay, not just complete stable, relatively stable micelle, which has a much higher drug load. That is the goal. Actually, what happens, if you remember the thermodynamics, I'm talking about the undergraduate students and the MSc students, the size of the micelle and the drug load or the number of drug molecules inside the micelles is governed by the Gibbs free energy of the corresponding unit in a particular system, if you wanted to make it by thermodynamics. So it basically, there are a lot of factors. Okay, so what is the nature of the molecule, what is the concentration, what is the solvent, what is the shape of the lipid molecules by which you are making the micelle. So there are so many things that governs the drug payload. But can we basically bypass all this and make a kinetic model by which we artificially increase the drug load? That is the goal. Okay, so what we did here, we choose a lipid has a dual head and a single tail. Okay, this tail has to be hydrophobic. This is otherwise it will not make a micelle in water. Okay, you know why? Otherwise it will, if this is a hydrophilic part, then it will form a reverse micelle. Okay. Okay, and so what we did actually, we take a nanotube. Okay, actually the water, this nanotube is inside the water in the big system. It is actually more than 1 million atoms. It's a huge system, so that's why we have to go to the coarse graining. Okay, so we put all the lipids inside the system, and then we allow them to absorb on the nanotube. Okay, so then they allow, and we inject or pump this drug inside the nanotube. Okay, and then we allow this, basically then we vibrate or basically apply the force from the left hand side to the right hand side, and you can see that basically there is a formation of micelles, and probably you see the red color, which is the drug. Actually, even if you see this paper, we also calculate what is the thermodynamically stable micelle and what is the comparison between the drug load. And we see actually the kinetically, this micelle has more than 10 times drugs inside this micelle with a similar size. That is important. Similar size and much drug load. Okay, probably this is a little information. The micelle cannot have random size. Okay, otherwise it will not cross the blood barrier, or there are so many complications. So it has to be particular size, and it has to be also monodisperse. Okay. But what happen if you take a one tail and one head? So this one has a double head and single tail. Okay. And this one has a single tail and single head. Okay. So, and you can see that it is not going to form micelles. Rather than forming a micelle, it is formed like a bicelles, like a tubular cells. It will not form micelles. So it is not good for the drug delivery. So by just doing a computer simulations, you can tell the experimentalist what kind of molecule you need in terms of chemical compositions, because that will dictate the micelles in a particular solvent, and also what is the architecture of the molecule that you might need in order to make these micelles. So there are two lessons. One, what is the chemical compositions of your molecules, and what is the architecture of the molecule that can give you the best micelle that you want. Okay, so if you see the movie, I have a movie, probably the red color is not very visible to all of you, and you can see how the micelles are forming from the tip. Okay, there are so many explanations here, but I do not have enough time to discuss it. And you see, the first micelles is bigger, and the consequence micelles is not smaller. This is because we do not have enough micelles, not the micelles, enough lipid molecules, and the force is constant. So that's why, as we go along, the size of the micelle will be smaller and smaller. But in a real system, when you have the continuous supply of the lipid molecules and the drugs, in principle, it should form same size of micelles, which is required actually. That is also called monodispersed particle. Okay. So that's it. So what you did here, actually, this one helps the experimentalist. Okay. So that the lesson is here, we simulate it here, we try to understand what are the principle, what is the process that we need to learn, and then we basically make a model, we try to explain these things, and then we deliver to the experimentalists so they can design their molecule. Okay, so this one is the real collaborations, and this is actually now FDA approved. I'll see you. Okay, so if you see this, probably dendrite and dendron, those are organic chemists. Okay, so these kind of molecules are now very famous for the drug delivery carrier. Okay, they will easily form the micelles because of the head to tail ratio, volume ratio, which is also a very important parameter in order to make a micelle. Okay, so one of the key parameter is the head to tail volume ratio. The bigger the ratio, the bigger the difference of the volume, then you need less number of monomers in order to make a micelle. Okay, and the number of monomers needed to make a micelle, sometimes it is called magic number. Okay, so what they did, they tried different kind of molecules. Okay, and then they try to understand what is the best molecule for the drug delivery. Okay, and we try to simulate only 4, but also in experiment they did probably more than 20 or 30. Okay, so this red part is called PEG, polyethylene glycol. You probably all know that. Okay, and this blue part is basically the PCl. This is basically caprolactam, but we break the chain. Okay, this is a hydrophobic part, but there is optional. You have the oxygen, because in a body there is an enzyme. It has to cut it, otherwise it will be toxic to your body. Okay, so when you design this kind of macromolecules for a drug delivery, you also need to make sure what is the fate of this kind of molecule when its job is done. So this has to be very crucial, otherwise this can be dangerous. So basically there are some ester bond inside here, and there are so many ester clipping enzymes in our body which can chop it, and essentially it will basically can go through the urine. Okay, that's why it is safe. And PEG is also FDA approved, which is considered to be safe. Okay, so these are the basically block copolymer. Okay, so this is the long PEG. You see, this is a PEG 2K. This 2K method, the 2K is the 2 delta. Okay, and this is 16K. This is the molecular weight. That's why you see the long chain. Okay, the PEG. And this is 3.5K. So 3.5K. That means the tail length is same. All tail length is same. But now this one is basically linear polymer, and now here you have this dendron. This yellow part is the dendron. Okay, then we simulate this micelle. Okay, so then we simulate this micelle. So these are the 128 monomers is needed to make a micelle. Only 14 monomers is needed to make a micelle, and 10 monomers is needed to micelle. So what is the difference? This one is the linear polymer. There is no dendrite involved, no dendron involved. Okay, the dendron has different advantage. So one advantage is it basically creates this volume ratio difference. Another one, in experimentally, it is very easy to functionalize this dendrite part. Okay, mostly they do the functionalization in this inside this dendron. Also, sometimes they functionalize the peg part. We'll discuss later. Okay, and so this one, you see that if you see this the left hand structure, you see the blue part. The blue part is exposed, which is basically hydrophobic. So this is not good for the delivery because it does not like the blood. When you put a lot of micelle inside the blood, what will happen? The blood will like only the red blood, because blood only like the hydrophilic part, but it does not like the hydrophobic part. Immediately what will happen? There will be maybe many, I don't know how many micelle, there will be a lot of micelle, they will like each other, because then there will be interaction between this blue part and another blue part of another micelle. Then they will coagulate. So the drug will not deliver to your cell. It will be just sitting on your under the skin, because the blue part is exposed. Okay, it does not, it is not good at all. Then on the right side of the extreme, also this is a dendrite, but what we did, we increased the PCL chain length 14K. But because we increase the core volume, so the volume to core volume ratio between the head and tail is not so great anymore. Again, the blue part, which is the hydrophobic part, is exposed to the water or the blood. Okay, so this is also not good. Okay, but look at the middle part, which is ideal candidate. Okay, by the way, the G3 is called the generation 3. Okay, now these lab actually have G6, and which is actually FDA approved now. Okay, for the drug delivery. So now this one case, the blue part is almost covered. So that is only the red part, which is hydrophilic and which is exposed to the blood. So the blood actually like it. And because this red part is kind of, you see, if you see the poly-PEG part, they're kind of charged because there's a lot of oxygen. So the two micelle cannot really come close to each other. So they will be separate, and they also like blood. So this is an ideal candidate for the drug delivery. So now by computer simulation, you can guide the experiment. What kind of molecule do you need? Not only what kind of molecule, what are the molecular weight? For example, that PCL14K is not really good, although you need dendrites. Okay, so that's kind of this kind of help you can do to your colleague, to experimental colleague, and they can synthesize, and then they can do for the drug delivery. That's the okay, advanced work of this. This is on his advanced work. So then they modify this peg chain with a different functional group, because think about it, if the drugs wants to, if you want to deliver the drug, this micelle has to attach somewhere on the membrane at some point, either in cell membrane or somewhere. So there must be some kind of interaction between the peg and the cell membrane. Otherwise, it will not see that. Otherwise, it will always just moving around here and there. Okay, so they did functionalize with amine, acid, OMe, and AC group, and then they try to identify which one is the best and which one is bad. Okay, so but they could not know the answer, which one is actually why someone is good, someone is bad. So we identify some of the reason why someone is bad or someone is not doing anything. For example, if you have the amine, okay, if you have the amine, for example, in this case, this hydrogen of this amine group actually forms the hydrogen bond with the oxygen of the peg, because the peg is a long chain. Okay, look, peg is a long chain. Then what happens? The amine group actually, it is not on the surface. It is not on the surface. What it does, it basically buried on the surface, make hydrogen bond with the oxygen of the peg. Look at this. So you expect from your eye that amine should be on the surface of the micelle, but actually, actually it is not. It is under the surface, doing some kind of hydrogen bond, and the charge that you expect with this positive charge with the NH3 is missing. Okay, so you cannot just do blindly micellization, functionalization. You need to understand what is the chemistry and what is the physics of the surface. Okay, so that's how it works. The computer simulation works. Okay, so now moving to the next. So now moving to the next. So this one is a little. I need to explain what is the origin of this project. So I think I'm doing right. Okay, so this is an enzyme called catechol. This O is ortho and methyl transferase. So what it does? So catecholate. If you see this catecholate, this is a neurotransmitter. So in our brain, we actually, in our body, we produce this kind of molecule, catecholate or something like this. Okay. Also, there are some derivative also you produce. These are called neurotransmitter. Brain. This is actually the brain is using it for the signal transactions, signal transmission for particular purpose. So what this enzyme does, it actually does some bad job. Okay. So this is a CH3. This is actually a substrate called SAM. So, and this is a magnesium inside a octahedral environment. Okay. So this enzyme, this CH3 transfer to this O minus. Actually, before that, there is a lysine group which takes the hydrogen of the catecholate hydrogen. So if you remember the catecholate, you have two OH. Okay, so this is O and H. This hydrogen has been already taken by a lysine. So this is now O minus. So because this is O minus, at some point in this active site, okay, so the active site is okay. So just let me tell you what is active site. So active site where in the enzyme where the reaction takes place, that's the active site. The simple definition is that. Okay, so CH3 basically transfer from this SAM to this catecholate. Once the CH3 is transferred from SAM to this catecholate, then it is not catecholate anymore. This is like catecholate with OCH3. This is kind of phenol with OCH3, and this is not neurotransmitter anymore. Okay, so the body does not have enough catecholate to do some your own work. So then you develop all these motor symptoms. Probably you know this Parkinson's disease. You have this, something like this Parkinson's disease, and so then what usually people think about, can we have an inhibitor? So we need to have an inhibitor so that it can block this site. If we block this site for this particular mechanism, then we do have whatever the enzyme, whatever the neurotransmitter is sufficient to do its own work. So the purpose of this project was to develop an inhibitor so that the CH3 group cannot transfer. Okay. We actually developed several molecules in order to find which one is the best molecule. But before do that, we did some molecular dynamic simulation to understand what is the mechanism. Okay. So if you see, this is octahedral environment. You see, this is a different residue of the protein. This is another residue, and this is the metal. So 1, 2, 3, 4, 5, 6. So this is a pure octahedral environment. Okay. So our goal is to design a molecule which can hold this place. Okay, like a keeper. So the active site of the enzyme is blocked, and no more catecholate is destroyed by that CH3 addition. Okay, that is the goal. Okay, so when we simulate this one, after some times we find that actually this hydrogen, if you see this hydrogen, actually flips to this side. Okay, you see this hydrogen, which is initially this oxygen and the metal was bonded, but now this hydrogen actually flips. So it's a dihedral flip. When the hydrogen dihedral flips, then this hydrogen of this catecholate actually form a intra-hydrogen bond within this O minus. Because of this guy is not anymore in this octahedral environment. So there's a new guy will come. So this kind of molecule may not be good for an inhibitor. So you need to have some kind of derivative of this molecule for a particular job, not for this. So, and we actually identify what is this problem. If you see after some time, okay, 18 nanoseconds, there is a flip-flop. That means this octahedral environment is destroyed. So once it is destroyed, that means you cannot take this kind of molecule as an inhibitor. Okay, we need to find something else. These are some distance between the bidentate and monodentate. By the way, this is when it is perfectly octahedral. This is bidentate because metal has a 2 bond with this catecholate. One is with purely O minus and another with the O of OH group. Okay, but when it is only O minus, not this hydrogen is anymore involved, and then it is a monodentate. And you can see after some time it flips. That means the active site is destroyed, and this catecholate guy is not bidentate anymore. This is monodentate after this. Okay, and then we did some amyloid sampling. Okay, so this is amyloid sampling means, okay, amyloid sampling is like a computational technique to understand what is the activation energy for this flipping, this dihedral flipping of this hydrogen from one side to another side. So this is, think about like imagine your Arrhenius plot that you need the activation energy plot, but whatever you see in Arrhenius plot is a one-dimensional plot. Okay, in one side you have a reaction coordinate. This reaction coordinate in this case can be dihedral angle, okay, or any bond. On the other side, you can have either the enthalpy or the free energy. Okay, but in this case, this is a two-dimensional lumbar sampling plot. Okay, this color basically is the energy. It's not single dimensional anymore. Okay, so something like this. You see, this is like RNAs plot. This is a single dimensional plot. Okay, but this is a 2D. So where we calculated the dihedral flip with the O is magnesium OH distance. So we identify who is responsible for what. And then we modify the molecule so that it does not flip. Okay, so but there are so many calculations under this, and you need also some machine learning in order to understand and in order to get the exact features. Okay, for example, so you also did some quantum calculations. Okay, so this is also called enzyme catalysis. Or enzyme catalysis reaction, right? Because there is a CH3 transfer. There is a CH3 transfer from the SAM to your O minus. So we calculate this energy of this transfer from this SAM or CH3 to O of this catecholate, which is around 20, let's say around 22 kilocalorie, which is actually quite good. This 22 calorie is not so easy inside the body, but body has a different way to do it. If you do this reaction 20 kilocalorie, it will never happen. It will never happen in reality. This 20 kilocalorie is really a lot. Okay, think about a KT in normal room temperature. It is 0.6 kilocalorie or something like this, and this one is 22 kilocalorie. Okay, but in our body, it has a different mechanism. Even this is 22 kilocalorie, it actually does happen. Okay, this is a cooperative effect and different thing. And also we use to understand what are the contributions from a different residue of the protein. And this is also called the graph theory. Probably do not know all this at that point, but you can see from this color contributions, these are the energy contribution from the different residue which are surrounding the active site. Okay. Okay, so then actually I don't think we have these slides here. So what we did now, the next part of this project, actually now what we are doing here, I am not sure the undergraduate students are kind of aware about machine learning, although probably this is a fancy word these days. Okay, maybe artificial intelligence is a fancy word. Okay, so machine learning is part of the artificial intelligence. If you want to design a drug, actually, you do not really need to do a lot of experiment these days. You go to a computational laboratory, ask them, these are the protein, this is my target, and the computational chemistry can give you filter set of atoms. Okay, all that means actually the computational chemistry can find a set of molecules from the entire chemical database, or sometimes called chemical space. Okay, and this is now more prominent or more actually important now once we have the machine learning techniques, where you do not have to do really all the simulations. You just know the features of the molecule, you just know features of the protein, and then you can develop a program. And once you have the program, you can find the millions of, you can find let's say set of molecules from millions of molecules. So from millions of molecules, it can give you really maybe 50 molecules. And then you can go for this 50 molecule experiment. Although, I mean, experiment level, they do a lot. So those who do experiments, this is also sometimes called high throughput scanning in terms of drug delivery or drug designing industry. This is called high throughput scanning in experiment. And in computer, they are also called virtual scanning. Okay, so now these days they fast go by virtual scanning. They don't do randomly like 50 years ago they used to do. As they do virtual scanning, and now the virtual scanning is much more powerful because of this machine learning unit, a machine learning thing. Probably you know this COVID, right? So most of the vaccine and as well as the small molecules are now fast designed by machine learning techniques. Okay, so this is the advantage of the machine learning theory. Okay, so now the last part. So how much time do you have? I forget it. Hello?
Niladri, you are almost over.
Oh,
but okay, we just continue 1-2 minutes. You can just.
Okay, so this is the important thing. So probably we know all about the drug resistance, right? So most of this case we know this is a malaria drug resistance or something. So one of the mechanism by which the drug basically they call resistant is called efflux pumps. Okay, in the undergraduate program they probably have the sodium potassium pump. So this is the same. There are several kind of pumps in our membrane. This is one of the pump is called efflux pumps. Okay, so this is called efflux pumps. So when you put the drug inside the membrane, inside the cell basically identifies that this is a foreign body, I don't like it. Then there is a particular pump designed for this kind of foreign stuff to exclude or expel from inside the cell from the outer cell. Okay, so what happens is over the time, the cell is so smart. Okay, if you put a drug, the cell will identify immediately this is a bad guy or foreign stuff. It will immediately open this channel, that drug will go out. Okay, so this is exactly the reason that inside the cell you will have a very little number of drug inside the cell. So that means the drug is basically losing its power because it does not have enough molecules inside the cell. So now actually this is an alternative way to design a drug. Now these days, actually, if you see, even for the COVID, they don't go for the new drug. They're looking for the old drug, because to design a new drug, it takes almost 10 years. And not only that, there is a huge problem of the safety. Okay, so now what they do, instead of designing a new drug, they identify where is the leak. Okay, and this is one of the leaks. So if you can design an inhibitor, or that means if you can block this channel for that particular drug, then your drug will stay inside it and the drug will do its job. Okay, so then it is not resistance anymore. Okay, so now we are working to understand what is the mechanism of this pump. Okay, this is called MEXB pump. Okay, and it has a three part. Actually, this is one part, this is second part, and this is third part. These two parts actually anchored with this outer membrane and the inner membrane, and this is with this periplasm part. Okay, so this protein, actually now I'm talking about only this blue guy. Okay, so this is only the blue guy. So this is a trimer protein, and each trimer protein has a particular pocket. So this is one of the mark. Okay, so this trimer, and this is one of the mark. Inside it, it has a channel. So that the drug basically comes from the, let's say this is inside the cell. The drug comes here, it goes here, and then it has a particular rotational mechanism by which it will go to this place. And then there is another mechanism, rotational mechanism, from here to here, and then ultimately from here to here. Okay, we try to understand what is the mechanism of this rotational behavior, and can we have a molecule so that we can block this rotational mechanism. Okay, so this is a schematic program. Okay, so then this is very standard. Okay, so but this is a different pump, and we kind of believe that this could be the same mechanism because it's the same class. So this is called loose. So these are the configuration of different MAR. Okay, so 1 MAR, 2 MAR, trimer. So 3 protein, we have 3 protein. At a time, 3 protein have 3 distinguished configurations or conformation. One is loose, another is tight, and another is open. And they work all together in order to expel the dark from the cell to the outer part. Okay, so we did a lot of simulations. Actually, this is one of the patented molecule, and we understand what is the special about this drug. And we also simulated the precursor of this, the previous generation of this drug, and we understand why it is good right now. Our goal is to understand what is the nature of the pocket and as well as the molecule, so that we can use machine learning techniques to design a new molecule. But in order to get the machine learning features, you need to do the molecular dynamic simulation to extract the feature. Okay, so we did actually, then we see actually that inside this pocket, it have enough space so that this guy can flip. Okay, this guy can flip. Okay, so now actually this is for the undergraduate students. Inside the drug, actually most of the drug actually, it interacts with this active site dust by van der Waals or hydrogen bond, and also sometimes plays a role and also electrostatic. But van der Waals has electrostatic origin. Okay, it's a fluctuation of electron. And we see actually there are 2 different configurations inside the pocket. Okay, for the same molecules. And actually, what happens by flipping this dihedral, it's actually enjoy more hydrogen bonding. That's why it has more strength to block that particular part. This is not a drug, this is an inhibitor. Okay, but the previous generation of this molecule has only 1 part, so it enjoys less number of hydrogen bond. This is the advantage of having this. And we also calculated what is the energy for the flipping to understand, because this energy is needed for the machine learning features. So, and this is it. Okay, so these are the all details. I think I should stop. And this is actually quantum calculations. So there is another way to do it. It's called quantum mechanics, molecular mechanics. So this is with classical. This is like a marriage between classical and the quantum, but this is complicated for the undergraduate student at this point. Okay, I'll stop. And this is my group. These are the two MSc students. They are doing PhD some other places. Okay, this is my first PhD student. And this is my second PhD student. So he's in second year, and he's in third year now. And he just finished it. I mean, he just completes his second year, and he is still in second year. Okay, thank you very much. So if you have any questions, this is our beautiful campus. So if you have a chance, you can always visit. You're welcome. Okay, and this is actually heritage building. So if you know this, this School of Mines is one of the oldest college in our country. It's 1926. Okay, it's very old, and this is a heritage building. And inside this building, there is a museum, and it has all these different kind of metal and also the minerals, which is open to public, and which have been found under the coal block. Okay, so probably you'll never see somewhere else except this place. Okay, thank you very much. So if you have any questions.
Thank you, Niladri, for your wonderful
presentation. Okay.
And
any questions?
I think, yeah, Supratim, I think you have collected some questions. So please forward it to Niladri.
Hello.
Yes, I can hear.
How can detailed molecular information can be obtained using the trajectory that is being governed by the laws of classical mechanics?
What is it? Okay, so okay, as I told you, if you know the position and the momentum by classical, for example, if you wanted to know what is the enthalpy, right? Or what is the binding energy? So you can always, because we know the potential, you know who is interacting with what, I can always gauge the interaction energy from the trajectory. Remember these equations? Okay, for example, is it shared? No. Let me share it. So for example, okay, so the trajectory has these informations. Yes, the trajectory has these informations, right? Every time in the trajectory, you have the R and the P. Think about this equation. You only need R in order to calculate the energy. For example, if you wanted to calculate the bond contributions, you know what is the standard bond from the spectroscopy, and from the trajectory, you calculate what is the R in your simulations. In the angle, think about the angle. The angle is nothing but a component, basically two vector. And the two vector is nothing but the T positions. Okay, similarly for dihedral, even if it looks like a dihedral, but actually it is nothing but a position of 4 points. So once I have the 4 points, I can have 3 vectors, and I can calculate the dihedral. And once I put this in equation, you'll get the energy. Is that clear? Hello?
Okay, we can go to the second question.
I mean, is that clear? So you see, this is the equations. Every point in the trajectory, you are recording the position R. And think about this equation. This equation has only R. And the rest of them are standard. Okay. For example, the charge on the particle is fixed. So in this equation, if you want to calculate the electrical component or the Coulomb component, you just need to calculate what is the distance between two atoms. And this is just a R, and R is precisely written on your trajectory. Okay.
Okay. So we go to the second question. Are the size or nature of the drug factors for the micelle formation and delivery?
What is it?
Are the size or nature of the drug factors for the micelle formation and delivery?
Yes, of course. It's a combination of both. Think about it. I have a drug which is not hydrophobic, but in between. Okay, in between. Then the drug will not reside inside the center of the micelle. Rather, it will sit on the corona of the micelles. Okay, so there is a fine balance between your drug, is the hydrophobicity and the hydrophilicity. You cannot have both. And there is a danger to increase the hydrophobicity. If you increase the hydrophobicity, then it will not interact with the protein. If you have more hydrophobicity, it will not form any hydrogen bond with the protein. Think about protein in the active side. I just explained in the active side, the drug interacts with the hydrogen bond and the electrostatic part. But on the other way, if you increase if you put all of them are hydrophilic, this is also bad because the protein has also some hydrophobic part in the side chain. For example, lysines, all this stuff, valine. Okay, you have also this phenylalanine. So this is also hydrophobic part. So there is a basically pi-pi stacking also is important for the drug interactions. So when you want to deliver a certain drug, you need to first know what is the drug, and then you devise accordingly what would be the micellar monomer of the micelles. Oh, I cannot hear you. I cannot hear you. I cannot hear you.
Yeah.
Yes.
What is magic number in this case?
Okay. Magic number what?
What is magic number?
Oh, magic number. Magic number, you know, this is like a magic number, quote unquote. Okay. Magic number is also different sometimes in the crystal too, in the nanoparticles. So in this case, they say the magic number for a particular monomer, with a particular chemical composition, also with architecture. When I say architecture, it is the volume to tail ratio. The minimum number of molecules is needed to form a micelle. It's called magic number. So let's say if I need 10 monomers to form a spherical micelle, so 10 is a magic number for the particular compositions. If I need 20, then 20. Okay. This magic number is also defined in a nanocrystal or in the nanoparticle system. Okay. Sometimes if you see the metal clusters, so the magic number is also defined for that too. So the number of atom which form the particular size or particular shape of the crystal or particle is also called magic number to them. So the magic number is defined in a different way on each field. Okay, in this case, it is the number of monomer which is required to form a micelle for a particular composition and shape. Of course, the solvent is important.
Okay, so the last question is how the drugs get attached to the micelles.
It's just that's an interesting question. Think about it. So that's why the drug has a balance. The drug is not completely hydrophilic. If you put the drug in the water is completely hydrophilic, right? The water does not like the hydrophobic part. So what basically then drug is smart. It says, okay, rather than inside the bulk water, can I go inside the micelles? Because inside the micelle, it can enjoy the hydrophobic interactions. Okay, and this is just nothing but a diffusion mechanism. It's just a diffusion mechanism. Okay, it's just a diffusion. If you put a lot of molecules, okay, a lot of molecules in the water, eventually it will go. Okay, that does not mean all drug will go to the micelles because there is equilibrium. That's actually that was I was explaining it. It is the thermodynamic equilibrium that gives free energy of if individual drug matters. The Gibbs free energy of the drug inside the micelle must be same with the Gibbs free energy of the drug outside the micelles, and that defines the equilibrium. When it is fixed, then the concentration of the drug outside the micelle and inside the micelle is fixed.
Okay, so all the relevant questions has been covered. Thank you very much.
Okay, so you're good.
So thank you, Niladri, for a wonderful session, and you have covered a variety of subjects, starting from the molecular dynamics application in water, graphene interactions, and various things. So
I skip the mathematics part because mathematics is boring. I kind of realize that, especially when you do this Zoom, the mathematics is also very hard to explain.
Yes.
Actually, and also the coding part.
Yeah, and also without having an eye contact, it's very difficult to deliver lecture.
It is very hard.
So we are about to conclude today's session.
Okay.
That was a very nice session. So before that, I request our head of the department, Dr. Chandrakanta Bandyopadhyay, to please deliver the customary vote of thanks.
I also see in the YouTube channel, the maximum number 175.
Oh, that's great.
175. I mean, now it is 100. So maximum was 175.
Yeah, that's a good number.
You see?
So, customarily, I like to express my sincere thanks and gratitude
Thank you very
to
much.
all the speakers.
So now I think all we can probably come to the video mode, huh? So that we can see each other.
Yes, I can see you.
Yeah, no, I mean all the speakers.
Okay, all the speakers. Okay.
Yeah, because this is a vote of thanks.
Okay, thank you. So on behalf of our college, on behalf of our department, and on my own behalf, I like to express my sincere thanks and gratitude to all the speakers for their nice deliberations and for staying with us for the long time despite of their busy schedule with us online. I like to express my sincere thanks and gratitude to Principal Maharaj for his keen interest in arranging this seminar, in web seminar, that is webinar. And I must thank Dr. Supratim Shui and Dr. Subhobroto Banerjee for their active participations, and also Saptarshi Chatterjee, who is acting behind the screen nicely. I must thank them. And lastly, I like to express my sincere thanks to all the participants, all the delegates, for their patient hearing and for their participations and questioning, brilliant questioning. I must thank them, and I wish success for all of them in their life. With all this, I thank you all once again. Thank you.
Thank you very much. Okay, should we disconnect, or is there anything to discuss?
Subhobroto?
No, I
Yeah.
think, sir,
Please,
officially the session is over. So
Okay, maybe we can disconnect.
Okay.
Maybe we can disconnect the student. Maybe we can disconnect the
Okay.
student.
Yeah.
Yeah. Now disconnect the students, Supratim. So,
Okay, so shall I disconnect?
yes.
Yeah,
No
yeah.
problem.
Thank you, man.
Thank you.
Thank you, Debasis da.
Okay, thank you.
Good.
মহারাজ এসেছেন। মহারাজ স্পিকার অন করা নেই।
Card.
মহারাজের স্পিকার অন করা নেই।
Actually,
এটা বন্ধ করলাম, না হলে তো সবাই দেখবে কথাগুলো, বাংলা কথা শুনতে পারবে, কারণ ইন্ডিয়ার বাইরে থেকে তো সব অনেকে দেখছে।
oh, no, I think YouTube channel should be off now, no? I can see you're still in YouTube.
হ্যাঁ, না, ইউটিউব বন্ধ হয়ে গেছে। 20 সেকেন্ড ল্যাগ আছে না, 20 সেকেন্ড ল্যাগ, তো ওই জন্য বন্ধ হয়নি। এবার
I'm giving some answer to the students on the YouTube.
ল্যাগ আছে 20 সেকেন্ড। এই যাক, খুবই ভালো লাগলো। বেশ, ভেরি গুড।
ওকে স্যার, যেটা বলছিলাম যে আপনাদের এমএসসি যদি স্টুডেন্ট ভালো থাকে, মানে যেটা পিএইচডির জন্য আর কি, তো এখানে আমরা ধরুন বছরে 10 থেকে 15টা মোটামুটি 10-15টা পিএইচডি স্টুডেন্ট ইনস্টিটিউট ফেলোশিপ দেয়। আমাদের এখানে রড়া থেকে আছে ছেলেরা।
না, নেট ছাড়া কি হয়?
হ্যাঁ, নেট ছাড়া হবে না, গেট থাকতে হবে।
নেট বা গেট
থাকতে হবে। নেট ইজ নট রিকোয়ার্ড ম্যান্ডেটরি, বাট গেট থাকতে হবে।
গেটটা তো পাই, নেটের সংখ্যাটা কমে গেছে।
নেটের সংখ্যাটা কম, তার কারণ টোটাল নাম্বারটা হয়তো একই আছে, প্রচুর ছেলে।
হ্যাঁ, যাদের এখন নেটের সংখ্যাটা আমাদেরও খুব কমে গেছে।
আর গভর্নমেন্টের টাকার অবস্থা তো খুবই খারাপ।